All right, here we are. I am with a friend, Ian Chen. I'm Donny Budinsky, and we are going to be discussing arguments made by Ian. Ian, you're kind of a legend in the evolutionary community, aren't you? So,
Am I? I don't know. Maybe in my own mind, perhaps,
in
Donny.
your own
But
mind, I'm
no,
sure in
just
at least
just
one
a joke.
other
Just joke.
person's mind.
Yeah.
Ian, how you doing
Yes. Yeah.
today?
Not bad.
Thanks for
Thanks.
doing this.
Not bad. Thanks. It's 8 or 9 p.m. here, so I know it's kind of early morning for you. So hopefully you had a cup of coffee and maybe get some energy.
Hey, it's a privilege to have you on for a podcast.
My
Podcast
first one.
discussing.
First one with you, Donny.
Well,
Hey.
we're going to be discussing several of your arguments in
Yep.
your debate, your end game debate with Dr. Dino. That was a lot of fun. You managed to get, I should say, we managed to get, since I moderated all three, an entire debate trilogy that is
Hmm.
right up there with your Lord of the Rings trilogy, your Dark Knight trilogy. So it's right up there as one of the best trilogies out there. We might just turn it into a DVD box set, Ian. What do you say about that?
Yes, I think, and I think, was I wrong or did I hear incorrectly? Kent was open for further debates. It surprised me, but if that's what he wants. So what I try to do, Donnie, is I try and think through some arguments that your audience may not either be familiar with or something new and innovative. So I've got a couple that I'm kind of still in the back of my mind. So if I can kind of flesh out a little bit further, we can either do another one with Ken, or you can set me up with another yet to kind of try it out on, basically. So
Yeah.
yeah, I like to try and do something a bit different. I know today we're going to be focusing more on the genetic side of the arguments that we kind of brought to Kent a few days ago now. So we'll do that, but I wouldn't mind at some stage also talking through the what I thought was an interesting take in the argument, which was the kind of two purpose. One is to kind of rile Kent up a little bit, but to kind of also say, hey, I'm going to try and argue that the Bible supports evolution, and that was what I thought would be something that was a little bit interesting and different. So maybe at some stage we can talk about that, because I still think that is interesting, and I do think that you can maybe argue that particular case. But yeah, let's focus on, because I know you're kind of, this is your house here. Let's focus on the genetic side of the last two arguments I brought to him a couple of
Well,
days ago.
you definitely have an interesting approach. And because of that, you make for some memorable, some engaging debates. I would say all three were very memorable, very engaging. You took, in many ways, a different approach each time, which kept it new, kept it novel, kept us guessing in terms of what arguments and strategies you're going to utilize. And so that's why, yes, we're going to focus largely on the genetics arguments that you brought up and discussed with Kent. Now, I moderated the debate, so I am not going to say who I think won, who I think lost. What I'll say is it was a great debate. It was a must watch debate, I would say. Both sides brought up some fair questions, some fair points. You guys had some solid back and forth. It was lively at times, but
Hmm.
that's to be expected in a debate like this. So, Ian,
So
I've got
are you gonna break? So in private, will you break the bad news to Kent then? Let him know that unfortunately he lost. I mean, it's okay. You don't have to say it in public, but you and I know, so you can break it to Kent in private. No, I'm joking. I don't know what Donny thinks. I'd like to think that he thought I did quite well, but I have no idea what he thinks. But yeah, I think it was. I think, look, to be honest, like I've actually spent a bit of time today just reading through some of the comments and stuff, and I'll try and address some of that hopefully in the discussion today. But I think, and correct me if I'm wrong, but I think some of the reason why your audience enjoys watching Ken and I is because it's lively. Because and people, reading the comments, it sounds as if it's just me that's throwing Ken jazz, but he does the same as well. Water off the duck's back. I don't really care, and I think he's okay. Like, I saw him kind of smiling as well. So it's a bit of that banter. It's a bit of that back and forth. The audience might not like it, but I think Ken is a big boy now. He
I
can kind of
think he
take it.
even invited you to Dinosaur Adventureland for a live in-person debate. So there you go. There's an honor.
Or I mean, who knows, right? It could be just a personal touch there. So
Maybe
who
to
knows?
take you out. Well, you know what? I'll go with you. I'll moderate. I'll make sure that you're safe and sound, that it's a good
Yeah,
debate.
yeah.
And you
Bring my hit
guys,
back to my wife.
well, the thing is, you guys have good banter. You're both
Mhm.
quick-witted. You're quick on your feet. You're good with rhetoric. So I think that's why you guys give stimulating debates. That's what we'll say. And so, Ian, I've got 2 goals in this podcast.
Yep.
Convert you to creation,
Hmm.
and I'm gonna convert you to liking DC over Marvel. Okay, so
All
those
right. But I
those
do
are
like
my
DC.
2 goals tonight. But do you like
That's right.
DC more than Marvel?
Well, Superman's my favorite. So if you
Okay,
pick a superhero, like I think you should know this. I like Superman. He's got a good story. He's a nice guy. He's also super powerful.
that's right, yeah.
So yeah, so I like Superman. So I don't, if you
Nobody
call
in
that,
Marvel can take on Superman. Nobody in Marvel can match up to Superman. Not even Thor, right?
well, I mean, if they're covered in kryptonite, maybe I suppose.
It's
But you
true.
know, every superhero has to have a flaw.
No kryptonite
Uh,
allowed.
Yeah, but he's powerful. He's super powerful. Right? Nothing can beat him. So yeah, no. So you've already done that. You've converted me to DC
Okay,
already. I prefer
that's true.
DC.
We've had some private talks on that, so that's good. I'm glad that we've gotten somewhere there, which means I've only got one more goal to get you to creation. So
That's
let's
gonna
get
be
into
hard.
the fun, Ian. I think you wanted to kick us off with something before we get into
Yes, yes.
your first genetics argument.
Maybe share my screen if that's okay.
Sure.
Right. Okay. So, and the reason why I'm kind of bringing this up is when I read through the comments, not just on the third debates, on the one that we did a couple of days ago, but just on basically all the debates I've done on your channel. Um, Apart from a couple of people, like with T Rock, for example, and I think I did one with a guy called Benjamin White. I think I actually don't have anything against these guys. They're actually fairly, whilst I think they're wrong, and I think they're very wrong, they're actually quite decent people. So you'll hear in those debates that I'm not insulting them. I'm actually trying to speak to them personally, etc., professionally, etc. So but with Ken, I just don't like the guy. So I'll kind of leave it there. But I hear a lot of people in the comments saying that I use a lot of ad hominem attacks, and I've said a couple of times I don't think creationists understand what ad hominem is. So I think I'll spend maybe 2 minutes just to explain it to your audience, and maybe they'll then learn the difference between what an ad hominem is, which I never do, and what an insult is, which I admit I do a lot, especially with Ken. So let
So,
me just
Ian,
flip.
what you're saying is you've had some fun post debate
Yeah.
reading the live chat and the comment section.
Yeah, yeah, yeah. So they're saying Ian does a lot of ad hominem. In fact, Kent does it as well. Kent says it all the time. So
You've got
look,
a lot
very
of creationist fans in there then, eh?
and the comments kind of make for really fun reading. So keep it up, guys. I love it. And in doing it kind of engages with Donny's channel as well. So you should do more of it. And I'm okay. Like I said, water of a duck's back. None of this kind of stuff bothers me. But the difference between an ad hominem and an insult is, let's say, person one makes an argument, and I've just pulled off this site and just kind of basically read it off here. So it's not that I'm making it up. You can look at Grammarly or other sites as well, and you can kind of read the examples. So let's say person one, the creationist, makes an argument that says, I'm tired of always being the driver whenever we go out as a group. Person two, let's say me, this is the ad hominem. And I would say, you're way too nervous about driving at night. So, of course, you don't want to drive. The key thing about an ad hominem is if you are making a personal attack on a person to counter the argument. So, the argument here is person one saying, he's tired of being the driver. And I'm saying, well, you're always a nervous person. And that's why you don't want to drive. That's the ad hominem. So, I'll give you another example that kind of highlights this properly. So an insult is when I say you're too dumb to understand the issue. The ad hominem is you're wrong because you're too dumb to understand the issue. You can see I've taken the dumb part, which is the personal attack, and related that back to the argument by saying because you are stupid, that's why you're wrong. All right. So I insult Kent all the time. I call him stupid and dumb. And you know what? He does it to me as well. He calls me an idiot. He says, believing in evolution, you're a fool and all that kind of stuff. So he's insulting me back and I'm insulting him back as well. But I do not use an ad hominem. Now, if you kind of go further, and here's a third example, if you kind of say you're wrong because recent data show that the issue is multifaceted and thus cannot be solved through a simple budget increase, you're dumb for using simplistic thinking. That's neither an insult nor is that an ad hominem because you are actually engaging with the environment. Oh, sorry, with the argument. Okay, so the difference between an ad hominem and an insult is with the ad hominem, you're using the personal characteristic of the person to say that they are wrong in their arguments. Okay, so I'll kind of stop it there because and I'll probably just flick to this. I'll finish off with this Wikipedia article that very clearly says here, and I think this is a creationist. A common misconception is that ad hominem attack is synonymous with an insult. This is not true. Although some ad hominem arguments, maybe insulting, which is obviously true. But yeah, I just wanted the audience to kind of know I don't do ad hominem. Stop saying that I do. Yes, I insult Kent, and I do it often, and that's fun. But you're wrong when if you say that I use an ad hominem. So I just
So
want to kind of clear that kind of. Yeah,
so
sorry,
what
go
you're
ahead.
saying is even though you and Kent, being the good friends that you are, you guys have
Yes, friends.
jabbed each other here and
Yep.
there.
Yep.
None of those jabs would be considered technically an ad hom.
Against Kent. Does that make sense, audience?
From from my understanding, homonym
Hmm.
is so ad hominem. Homonym is a Latin word that means to the man. So technically, an ad hom fallacy is actually, before I continue, Ian, on my screen, you're frozen. Are you?
I can hear you. Can you hear me?
I can hear you, but for some reason you're just frozen on. Let me see if I were to.
I will try and I'll stop screen because I finished with this anyway, and I will stop my cam and restart my cam. Nope, that doesn't work.
Yeah, you're kind of just frozen in
Stop cam.
a position that you're just kind of looking at the screen. Yeah, we'll have Ian drop out and then make his way back. Okay. Looks like we've got Ian back with us. Ian, I appreciate you breaking down the ad hom fallacy or ad hominem fallacy. Do I approve of any side, any of the debaters insulting, whether ad hom or not? The answer is no, but it does happen. And sometimes the banter makes for a more stimulating or engaging debate. And so it happens. But what I like is that you guys shake your hands you shake each other's hands at the end of the day, and you move on and you get ready for round 4. So that's good, Ian. And what I
Yeah.
was saying before you dropped out, from my understanding, homonym means in the Latin to the man. So yeah, technically, as you were saying, Ian, an ad hominem fallacy would basically be attacking the person's character, attacking the man rather than the argument itself.
Yeah, but it's
Yeah.
the key thing is using that as your rebuttal to the argument, right? So as I was
Right.
saying, just to
Solely
say,
as your rebuttal.
Donny, you're ugly is not an ad hominem. That's an insult for sure, 100%. But Donny, you're ugly, and that's why your arguments on ERV is wrong. That's the
Right.
ad hominem. It's
Right.
basically saying I'm not addressing the argument at all, except by using a personal characteristic of yours as my address, as how I address the argument. That's what the ad hominem is. It's a look as Wikipedia, as I showed on Wikipedia, many people commonly mistake what these two are. I just wanted to kind of
Yeah,
address
because
that for
on that Wikipedia page, it
yeah,
did make the distinction between an insult and an ad hom, that they're
yeah,
not synonymous.
yeah. And
And
guys,
so it would
I
be like
insult
you
Kent
saying,
a lot. 100% agree with that. I insult him a lot.
yeah.
That's fine.
And
Yep.
it would be like you, like you said, calling me ugly, calling me stupid, and that's why I'm wrong, rather than attacking the substance
Hmm.
of whatever argument I would make.
Yes,
So
exactly.
yeah, yeah. Okay,
All right.
well, Ian, appreciate that.
Okay.
And
No
so
worries.
let's pull it
Maybe,
up.
maybe share
And
the slides, and I'll quickly summarize. I won't take as long. I'll probably need a
now
couple of minutes.
I'll say
Take
this,
it.
Ian. I know you've
Yep.
heard this before as you're pulling up
Yep.
the slides.
Yep.
One major reason that
Hmm.
I like to focus on genetics when debating or engaging this topic and question of ancestry is because I strongly believe genetics, how it's a direct way to answer this question of ancestry, right? Genes and traits are inherited, sperm and egg. It's an
Hmm.
empirical way to say, hey, what's true? Universal or separate
Sure.
ancestry, rather
Sure.
than looking to fossils and historical reconstructions. Those things
Yeah.
are not inherited sperm and egg. Now we can look to those and engage those and see how that evidence fits the
Well,
debate.
they,
But
well, they
genetics,
cross
it's a
confirm
direct
in
way.
genetics. Okay, so and keep in mind, genetics came after what we kind of know about fossils, and it was
Right.
interesting that when we go back and look at genetics, it actually cross confirms what we learned from the fossil record, etc. So, I mean, the importance of the fossil record by itself, you're absolutely right, it's not as strong as the genetic evidence. However, you got to look at everything together. Look at everything that we've learned together, and you got to ask
Mhm.
yourself, why is it that all these different evidences from different sciences all seem to link together and cross confirm each other really well. So that's one, I think, of a bigger piece. Instead of looking at things individually, one of the arguments that's the creationist is why does it all fit together better in that evolutionary mindset rather than a creationist mindset? That's a point that I'll just make.
Yeah, and that would be a point worth engaging, and to not engage it for too long, just so we can get to your first slide here. I wonder though, Ian, and I'm curious as to your thoughts. Is that true? Because from my understanding, and correct me if I'm wrong, prior to the sophisticated genetic sequencing that we're able to do now, the genetic data that we have now, homology at a genomic level is not necessarily predicted or expected because they expected randomness. Because if we've been evolving for millions of years, right, we're accumulating mutations and genetic change for millions of years, then why are there such strong
Can
genetic
you expand
signatures?
what do you mean by we expected randomness? So forget was it in the 40s or 50s that DNA was first discovered by what's that Watson and Crick? So before that, we had even I think a creationist himself, Carol Linnaeus, started to use taxonomy to figure out where different species sat in the
Right.
record. Okay,
Yeah.
so then that's when he himself, I think, I can't remember
Classification.
yeah, the classification of life, right? That's when he himself said, please tell me the difference between a human and the apes, because when I look at the different characteristics, he didn't know about genetics, but so he looked at
Right. So
morphology
he
and
he was
anatomy.
looking at
Yeah,
morphology,
yeah,
anatomy, right?
yeah, and he came out, and so there was a nested branching pattern of what he said was, and then Charles Darwin came up with his view that everything came from a common ancestor, etc. And we built up a picture of different animals evolving with a common descent into these different lineages, right? So we even before we had DNA, we suspected that apes were our closest ancestor. So we could tell that because when you look at our morphology, we all had the same bones. Now it was maybe different sizes and stuff, and but we essentially had the same bones. Like a gorilla and us, we have the same bones. The shape of the back of the spine might be different, and that's why one of us kind of like walks upright, the other one uses kind of the hands and the feet to kind of walk, or quadrupeds. But we had the same bones, so we kind of look at the bones and kind of fit it in a nested hierarchy. Now then, we got genetics, and we were able to sequence. You know what I mean? Like, it confirms what I'm
Well,
saying.
right, but
What
but
we
but
knew.
my point is, yeah, they did recognize, like Linnaeus with
Yeah,
classification systematics,
yeah.
kingdom, phylum, class, order, family, genus, species. Okay, we can classify these organisms. They recognize the homologous patterns, the similarities in anatomy, morphology,
Mhm.
right? Okay, across the biological world. But did they predict that same thing on a genomic level? Like, as
Mm,
in when you would see the underlying blueprint, if
mhm,
you're accumulating mutations over time,
mhm.
then those homologous patterns, those signatures should be weak, but they're not weak. Once we perform these genetic sequencing, we discovered strong signatures. We found homologous patterns not only in morphology and anatomy, but also in genetics. And so I'd be curious to know,
So what do you
was
mean by
that
that?
predicted
Because
or expected?
To be honest, I don't know the history of what we predict. So I can only speak to what I have learned in terms of the end result. Whether we predicted or not, I'm sure we did at some stage, because that's the whole point about people doing papers and stuff where they kind of make a prediction and they test it and then they
Right.
publish the results. So I'm not sure about the history of the prediction, but what I can say is when you look at what we know about humans, so I'll stick on humans and chimps because we kind of talk about that later in my slides anyway. But when we look at humans and chimps and gorillas, and we've sequenced their genome and we kind of looked at how close that they match, it's not surprising that the chimpanzees and the humans are the closest. Now, I won't get into debate about whether you think it's 95 or 85 Tompkins. I think Erica does a really good job on explaining what Tompkins got wrong on how he got his number. But either way, it is still comparatively the most similar of the
Yeah, I,
animals.
I I wouldn't have a problem going with the 96% number
Or
because either
96
way,
or 85, it's still chimpanzees and humans are still the closest as
yeah,
well.
85, 88, or 96,
Right.
humans and chimpanzees would be the
Yeah,
the
yeah.
closest
I would
genetically
say it's 96, but
or
it's the
genome.
closest. And but if you kind of look at different mammals kind of going out to dogs and cats and stuff, you're going to see this difference increasing, which just so happens to fit the taxonomy that we had discovered that we had put together. And there might be a few kind of little bit of changes here and there as we kind of learn more about genetics, but most of it was actually there before the genetics confirmed it, right? So that if you can call that a prediction, that is like we're saying this is what the hierarchy looks, the nest of hierarchy looks like. This is what genetics show. Whether you want to argue whether it's 96% or 86 or whatever, it's still the closest, right? It still fits in that nest of hierarchy prediction. Now, I'm not sure if that was published somewhere in some paper to kind of say that was a prediction, but it kind of
Yeah, because
seems
I
to be
could be
right.
wrong, but I've heard at least one biologist point out
Yeah.
that it was to the surprise of the evolutionary community when they discovered such strong homologous signatures and signals in genetics,
Mm,
because they assumed, okay, if mutations have been accumulating, okay, for millions of years, mutations obviously adding the necessary genetic variation for selection to act
yep.
upon. I understand that. Then the signatures should be more scrambled, but they're not. They're strong. We do find homologous patterns in not only anatomy, morphology, but genetics. But that's something we can confirm at a later
Yeah.
time. That's my current
Let,
understanding.
maybe park that because that might be relevant to the fourth. So I brought four
Yeah,
points to Ken. I said the Bible proves abiogenesis, the Bible proves evolution. We'll park that one and we'll focus on the third and fourth point. So the third one being chromosome fusion 2, and the fourth one being the genetic
yeah.
markers, basically.
And
Yep.
and let me
I
say
think
this,
the point.
as we park this, I will say when it comes to Linnaeus, he
Mhm,
did classify,
mhm,
for example, whales. Okay. Yeah, he
yep.
classified them as mammals instead of fish. And he wondered, where is that separation with humans,
Mhm,
who you would call a human great ape, and then chimpanzees, bonobos, gorillas, orangutans as non-human great apes. Okay. So where's that line? Where can we separate
yep.
these creatures?
Mhm,
But he classified organisms based on function. So yeah, functionally or physiologically, whales diverge quite a bit from sharks. And it sounds like Linnaeus recognized that. And so that's why he classified whales as mammals, just like he classified humans as mammals, because
yep.
whales diverge physiologically and functionally from
Yep.
sharks. Even though they're both in the water, they
Yep.
do
So
diverge
I
strongly.
so the one distinction I'll probably make there is that for fossils, okay, where we don't have the extent animals to study today, you're absolutely
Right.
right. It's really going to be looking at the anatomical differences and making some assumptions as to what that. So one of the things that I heard today was interesting is, well, how do we know a velociraptor had feathers? All right. I don't think that we've actually found a velociraptor with imprints of feathers, but what we have found is what we call quill knobs on their ulna, so this part of the bone, basically. So the kind of arms, like they've got arms like this that we initially thought didn't have feathers because we thought they were claws, but then what we found was on this side of the bone, there were quill knobs where we know today the actual quill of feathers kind of attached to the bone. So we knew that they had feathers based on that evidence, right? So what I'm saying is that in the fossil record and anatomy and how it kind of relates makes sense, right? But when you look at whales or you look at, so the reason why we know whales are mammals is not just on the bones, but we look at its characteristics. So it
Right.
has mammary glands, for example.
Yeah.
It's the spine goes up and down. It's not this way in a fish. So it goes up and down, which is what we do as well. So we walk like that, whereas a whale kind of swims like this, right? So they
Right.
have the same similar vertebrae as this, and not as a fish. So the reason why we know the shark and the whale diverged so many years ago, now you would disagree with this, but we'd say hundreds of millions of years ago, is because the evolution of the spine would have been the first thing, because we say vertebrae are one of the first things, or chordates and vertebrae are one thing for the first things that evolve. So it's got to be way back in the side in the past where the different vertebrae kind of moves either side to side like a fish or up and down, right? So that's why we're saying that the diversion of whales and fish happened many, many millions of years ago. But we know it's a mammal because of the characteristics that we can see today. So when you say it's anatomical, definitely for fossils, but when we compare animals today, we can actually see more than just the fossils, right? We can see
Right.
behavior, we can see all that kind of stuff. Yeah.
And my
They feed
point
the kids.
is, Linnaeus wasn't necessarily confused as to what's true, special creation or
Mhm.
common ancestry, but when he was classifying animals, he recognized what you pointed out, right? Mammals differ significantly from fish, for example, where the females produce milk through mammary glands, suckle their young, breathe air, okay, have hair. And so functionally and physiologically, Linnaeus would not classify a whale as a fish.
Mhm.
functionally as a mammal, right?
Mhm.
So that's why, as a creationist, I would largely look to, and that's what we're going to get into here with your points on chromosomes, and then also ERVs, is we would look to nested hierarchies, because that's largely what you're pointing to, and I appreciate it, Ian. It's a good point. Nested hierarchies by design, as
Mhm.
compared to nested hierarchies by descent, which is what you'd say. That's why you made the example
Yeah,
of shark
and
and rope.
I think it's gonna come down to what you think is the more probable, all right?
Yeah,
Because there's always
parsimonious.
honestly, I've heard you talk about the design stuff, and you know what? On a surface level, it kind of makes sense, but from obviously as you get deeper and deeper, there are kind of questions that I would have for you. Same thing. You'd probably look at what I say, and you kind of go, I can see where Ian is kind of concluding it based on this evidence, but I've
Right.
got some kind of so I think the question for the audience anyway is like which would be the most probable based
Yeah.
on the evidences that we have. Now,
Well,
if
right.
I can quickly, I'll quickly start this so we can go straight to the. Well, I think we'll be able to nut out the chromosome 2 fairly quickly, and we'll go straight to
Okay.
the more interesting one, right? So,
Yeah.
um,
Feel free to screen
have
share.
I haven't shared yet. Ah, damn it. Okay, sorry, I thought I had. Okay, let me know when you have that. That's sharing.
Yes, there
Right.
we
Okay.
go.
So I'll quickly go. I'll finish this up in basically a couple of minutes. But I'm actually sometimes bemused as to why creationists really want to die on the hill with chromosome fusion. Okay. So I know Ken asked many times, what evidence do we have of chromosome fusion? Look, honestly, Kent's not a scientist. I'm not a scientist. Um, I've looked at some papers and stuff, and maybe I should have put the paper's name in, but would Kent look at those papers? I guarantee not. So my question is, whether chromosome fusion 2 occurred or not, whether you believe it occurred or not, I actually don't think it harms the creationist view. All right, a creationist could say, okay, if I was a creationist and I had to try and make chromosome fusion 2 work, I'd say, well, Adam had 48 chromosomes, which is the same as what we see in chimpanzees and gorillas. And because of his, what was it, the sin and the fall of man, it degraded. And then subsequently that became 46. Like, I don't see why a creationist would die on the hill and say chromosome fusion 2 did not happen. And what I was trying to show Ken is that, and I'll
But let me
skip a couple.
comment on that, because I think you do make a good point. We'll go one
Yep.
point at a time, and
Yep.
that's a fair point. Yeah, there's a lot of overlap in the creation versus evolutionary models. Okay, and so
Mhm.
there are a lot of lines of evidence. We all have evidence. How do we interpret it? Okay, well,
Mhm.
there's a lot of pieces of evidence that are agnostic, and I think the chromosome 2 is largely agnostic, okay, to the overall debate, because as you pointed out, humans could have been created with 48 chromosomes, as well as the great apes, or the original archetypes for great apes.
Mhm.
And then there could have been a fusion shortly after the flood, or shortly before the flood, because there was a bottleneck, and
Mhm.
therefore that fusion, that unilateral mistake, could have been passed on. So I do think that's a fair point, and so it's not a hill that we should die on, but I do find it interesting, and so I'd like to get your thoughts on this.
Mhm.
Is it agnostic for the evolutionary model, though? Because if there wasn't a fusion to explain the discrepancy in chromosomes, that would be a problem. That would be a point for the separate ancestry model, because what would be the alternative, right? If there wasn't a fusion, how would you explain the
Yep.
chromosomal discrepancy?
So this goes back to what I kind of alluded to before. Evolution for evolution doesn't rest on just one thing. So yes, if we just looked at chromosome fusion to itself and you accepted that it happened, you could, with no other evidence, kind of say, well, it's evidence for common descent, which is obviously what I'm trying to show that it is. Or you could say, well, Adam had 48 chromosomes like a chimpanzee and a gorilla, because God created it that way, and he sinned, and there was a global flood, which some sort of environmental factors caused some sort of rapid mutation of some sort, and therefore Noah and his descendants, fused the chromosome, and that's why humans today have 46. You could say that. And we couldn't really show if that's true or not either way, because we don't have Noah's genome, we don't have anyone but before that to Adam. So we couldn't prove it either way. So you're right. If
I
just
think we would, if I could, Ian, I apologize,
yeah, yeah,
because
go
that's a
on.
fair point. I think if we were to go with the fusion route, and just
Mhm,
say, for sake of argument, okay, we're not
mhm,
going to do this the whole time, but for this specific point, for sake of argument, there is a fusion. Okay,
mhm,
which model can best explain it? Well, as you know, there is a significant lack of what's called heterochromatin. That type of genetic content is completely absent in human chromosomes, but it is present in chimpanzees, in bonobos, I think gorillas. And so, okay, maybe there was a human specific chromosome fusion. I mean, you would agree, even according to the evolutionary model, the fusion would have taken place after the split in the human lineage, right? Your
Mhm.
hominin lineage. Okay. So if there was a fusion, I think the creationists could better account for it because we do have that heterochromatin discrepancy. If it was related to chimpanzees in any way, okay, then
Can I
we
can I
should
sorry
find some of that.
pause you
Yeah.
here, Donny, and this is and I'll just to be fair to the audience, I got a degree and a master's in business. So my degree and master's is in marketing. Science is something that I have an interest in, but certainly have not studied it. So, and I'm going to make a little joke here, Donny, which I know you'll get, and maybe some of your audience will get. But heterochromatin, could you maybe explain that for the audience? Do you remember?
Oh yeah, yeah. You're pulling a Dan Stern Cardinal, and I appreciate that.
No, no, no. It's. I think it was a was it Kent Hovind's debate with was it Conspiracy Cats when Conspiracy Cats was asking him all these terms and he said, "Could you explain that for the audience?" But it was clear he didn't know himself, but it was just trying to say, "Could you explain that to the audience?" So, look, I'm not really up to date with what that is. Could you maybe explain that to me so that I can maybe
Yeah,
just yeah,
yeah. Heterochromatin is basically this densely packed piece of DNA,
yep.
okay, that largely comprises repetitive sequences
Mhm.
like satellite DNA.
And
Okay.
why you're saying it's in the human, but it's not in the chimpanzee or gorilla?
Right. Yeah, there's a prominent paper out there that goes into the heterochromatin discrepancy between humans and the great apes, and it's not present. It's lacking in the human chromosomes, including the human chromosome 2, but it is present in the chimpanzee chromosomes.
And
And so
where
the
does it
question
sit
is,
on?
where did it go? Was it all lost? Was it all
Yeah.
added?
And look, for my limited knowledge of human chromosome 2, so it is a thing that's for human chromosome 2. It's not a generic thing that, it's just a genetic difference between Homo sapiens and the other apes. It's related to a fusion.
Well, if there was a fusion after the split. So let's say humans and chimpanzees, hypothetically, for sake of argument, let's say they're related. Okay,
Okay.
then after the split, your human lineage goes one way, your chimpanzee lineage goes the other.
Mhm.
Eventually, there's another branch, the bonobo branch. Okay, and then in the human lineage, there's a fusion event. Well, if that is related somewhat to the great apes, like the chimpanzees, bonobos, then there should be heterochromatin in the chromosome too,
Mhm.
but it's not there, and so
And
that
that
would suggest a
that
human
paper
specific.
did that paper give an explanation? I just don't know enough about this. Sorry for the dumb questions,
No, that's
but
okay. Yeah,
uh,
they
what did the
from what
paper
I
say?
remember, they proposed, I think, large scale deletions or loss of the heterochromatin in the human lineage. Because from an evolutionary starting point, you got your great apes that have the heterochromatin, right, in the chromosomes, but it's absent in human chromosomes.
Hmm.
So, what would be your most parsimonious explanation? That it was gained in all of your great apes, or it was lost in just the human lineage? So, if I remember correctly, they proposed that it was lost in the
Hmm.
human lineage. Have they tested that? I don't think so. I mean, how could they demonstrate such a thing, right? They wouldn't be able to show us when it was lost or why it was lost, but I think that's what they largely propose. But I could use that line of evidence to say, okay, there's a human specific fusion that happened sometime either right before the flood or after the flood, and
Hmm.
why it's unrelated at all to your other great apes is because of, and I could point to the lack of heterochromatin.
Yeah, yeah, okay. No, okay, that's on the face of it does sound like an interesting thing. They could very well have been a deletion if that's what the paper says. I haven't read the paper, but I'm sure they provided at least some hypothesis or evidence on it.
Well,
But when I say human
Ian, if
chromosome,
you don't mind, just for the audience sake, I wanted to largely keep it on your slide, but just real quick. So
yeah,
here's a paper that people can look up. the evolution of African great ape, subtelomeric heterochromatin, and the fusion of human chromosome 2, genome
Mhm.
research. So here's an excerpt from it. Chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin. So again, heterochromatin is that densely packed piece of DNA that largely comprises
What does a tandem
repetitive
satellite mean?
tandem satellite sequence. So
Yeah.
that would be your repetitive sequences, right? It's occurring in so you just got a lot of it, a lot of the same DNA, basically. Okay, so essentially, what this paper is saying, and the challenge to proponents of evolution, is why do chimpanzees and gorillas have these large blocks of subterminal heterochromatin, but human chromosomes don't? I would argue it's because they're not related. So the humans never had that piece of DNA.
Mhm.
But you would have to argue that, okay, at one point humans did have
Yep.
this kind of DNA, but it was lost after the split. So,
Yep. So yeah. So look, it's interesting, and I need to look this up as well. I don't even know what heterochromatin is, but my question then back to you would be, how would the creationists explain it? So obviously, God created this differently. I guess you'd say. Um, I mean, why is it different in humans? What does it do? I mean, I assume that at some point in Adam it would have done something. It's perfect. So it would God created humans with this missing. I'm guessing that you're saying it's a loss of it. So what, like, did it used to be there and it disappeared? Like, is that what happened, or did God create it missing because humans didn't need something that apes did? Like, what would be the creationist explanation for this?
that's a fair question. It's a good question. Since there are many similarities between humans and chimpanzees, okay, then we're going to expect a lot of similar genomic elements, genetic sequences, but there still are differences, as we'd agree, in anatomy, morphology, physiology. And so perhaps there's types of DNA, pieces of DNA or DNA units that one, like the chimpanzee would have, that's specific to its function and different to
So, so
humans.
what
So
would
it's
this
largely speculation, but we do know heterochromatin is functional. Okay.
what
And
does it
so whatever its function is would be specific to something like a chimp or a gorilla.
So what does it do for chimpanzees and gorillas? And look, I'm sorry for it's a basic question, I know, because I don't like this is probably the first time I've heard about it. I'm sure you've mentioned in your discussions, but it's definitely the first for me. What does it do in a chimpanzee? Like what I'm just trying to figure out. I'm just trying to answer your question as to why it's missing in humans. So if I can figure out what it does, like for chimpanzees and gorillas, then that might give me a clue as to why it's missing in humans. Does that make sense?
Yeah. Oh, yeah. It's a good question. Let's say it's involved or participates in the gene expression pathway. Gene expression being that process by which a gene is turned on to produce either a functional RNA or a protein. Okay, well, the overall morphology and anatomy, okay, which is coded for by the underlying genetics of the chimpanzee comprises a lot of differences.
Hmm.
Then I would argue, and I'm not sure exactly what, but that heterochromatin could play a role or a part in whatever those differences are
Okay,
in the
so
chimpanzee
okay, no,
compared to the
I got you.
humans.
I got
Yeah.
you. And I'll try and explain this. Keep in mind, audience and Donny, my fairly lack of understanding of this because it's pretty much the first time I've even discussed this with anybody. But if I were to think this through logically, there are two pathways here, right? There is God created it. Chimpanzees and if you're saying gorillas have it and they need it for whatever the gene expression is that you've mentioned, that's obviously something that's those animals required and humans do not, and that's why humans don't have it. So there's that one option. God created it that way because it was functional for those animals and we don't need it, so God didn't give it to us. Okay, so that's option one. Option two could be potentially humans and apes both had it, and maybe one of the reasons why humans no longer have it now is that whatever these gene expressions are that you mentioned that chimpanzees and apes do that humans no longer do, that sounds to me like it then becomes not fixed. Like if it's not something that humans require anymore, then that would not be a selective pressure for Homo sapiens to kind of keep those genes around, and mutations could appear or deletions could appear, and basically kind of remove it from the gene pool for humans. So I could argue it that way as well. But that's, like I said, based on the very 10-minute discussion that we've just had on a subject that I pretty much am starting from scratch, right? So I kind of see like there's two explanations for it. Now, as I kind of said earlier, it becomes a question of what is the probability of both explanations. You get what I mean, Donnie? Like
Right.
which one is the most probable? And if I kind
Yeah.
of go back to answer the first question from you, chromosome 2, I think, by itself doesn't point to the creation view as being the better view, or uh, the evolution view as being the better view, right? Because as I said, we can both make up or come up with examples as to why one or the other is the best explanation. But what I was kind of saying at the beginning of this conversation is there is a whole bunch of other evidences, or evidence, I should say. I keep on calling it evidences. Whole bunch of other evidence that you can kind of have to look all together, that then kind of say, well, one view actually can probably is best explained by all these evidence, and the other view can't be explained by all these evidence. And that's when you kind of look at all these different points, and you kind of say, well, evolution is the best view. And
It's the
so I kind of
network of
yeah,
evidence, you would
exactly.
say. Yeah.
And I agree with you. I'm not disagreeing with what you said, Donnie. When you just look at chromosome fusion 2 by itself, your explanation could be right. Okay. And my explanation could be right as well, but it doesn't really point. Like, I think mine points stronger to evolution, but I couldn't disprove yours based on chromosome fusion 2. And that's, I think, why I've concluded that I just don't understand why creationists want to die on the hill and say chromosome fusion 2 did not occur, when even if it did occur, it can point to creation anyway. Like, do you get what I mean? Like, it's not, to me, like a place you kind of don't, especially as we have evidence of chromosome fusion 2. That's why I showed the muntjacs and the deers before.
Chromosome fusions, yeah,
Yeah,
they
yeah.
definitely do happen.
It happened, and it happens in all different animals and many, many different species, and especially closely related species. So that's the key point. You can look at canines and dogs and foxes; they have different chromosomes as well. Same thing with bears. You look at panda bears, you look at the polar bear, black bear. I think one of them actually, I think the panda bear has a couple chromosomes less. So bears kinds as well is different. Horse kinds the same as well. Zebras, horses, they all have different chromosomes. Not every animal species has chromosome fusion that occurs. So then my question, which is an overarching question to Kent, was why then do you accept that for the other animals and not for human slash ape kind? Like I said, it's not a die on a hill for creationists. You can just say Adam had 48. There was something that happened in Noah's flood, something about the fall of man, or sin, and it became 46. You can say that. It doesn't disprove creation. But I just don't understand why Kent and some creationists probably on this call or listening now would want to die on the hill and saying that it did not happen with humans or apes. That's what I didn't
No,
get. That was my whole question to Kent.
well, a couple points, and again, there's some fair points for sure.
Mhm.
If let's say hypothetically there was a fusion, I'd be curious as to your thoughts here. So
Yep.
if humans had a fusion event, okay, it would be specific to the human lineage. It would have happened after the split. So largely it is agnostic. And I do agree it's not a hill we should die on. But if there wasn't a fusion, then it would be curious because of that discrepancy. What else could explain that discrepancy? But if there was a fusion, it's largely agnostic to the debate. Okay. But, and I'd like you to speak to this. So if humans had a fusion event, it would have had to be a near extinction event, right? So you got that one person in the past who, well, I should ask you. So after do you put the split roughly 6 to 9 million years ago? Is that where you'd put it or hold
So
to?
I think from memory, humans split from chimpanzees about 6-ish million years ago. And I think the gorilla split, common ancestor for human and chimpanzees and gorillas, was 3 million years before that.
Okay.
So you
Or,
say
or,
six
or-
and nine, it's like six with chimpanzees, nine with gorillas. That's what I can remember.
okay. So you would say the human and chimpanzee lineages split from a common ancestor, which
Yeah.
wasn't human or chimp, but
Yeah.
whatever that common ancestor was, they
Chimpanzee.
split 6 million years ago. It would have been chimp-like, you'd say.
It was a human Z, I guess. So
Okay.
no, I joke. Yeah, it's a common ancestor between both, whatever you want to call
Okay.
that.
So then there's a split. You get the chimpanzee lineage going one way, you get the human lineage going the other. So your human lineage would be your hominin lineage. That's where you'd find, according to the evolutionary model, that's where you'd find your Australopithecines, Homo habilis, Sediba, so on and so forth.
Mhm.
So At what point do you think after the split within that 6 million years, at what point do you think that fusion happened?
It's interesting. I actually am not aware whether we can actually date. Or maybe we can. I'm just trying to think whether what I from what I know. I know we can possibly the reason how we date the common ancestor between a human and chimpanzees. I'm pretty sure we don't necessarily look at chromosome fusions. We look at the genome. So we look at ERVs, we look at ALUs, we look at lines, we look at other parts of the genome that we can see as shared with chimpanzees, and we do molecular clock dating and come up with a good guess on how long it takes for mutations on those particular shared parts of the genome to take, and we kind of work backwards and kind of go, well, it's roughly about 6 million. So we might look at how that works with ALUs and how that looks with ERVs, and it kind of converges to a 6 million date. So I'm pretty sure that's how we date the common ancestor between
Right, no,
chimpanzees.
I understand that, but the fusion itself, you
Yeah,
would
but that's what
agree.
I'm saying. I don't know whether
Okay,
we can
okay.
date the actual fusion event. It we know it had to happen after the common ancestor or after the split between human and chimpanzees.
Do you think
And
it would have occurred? And I know a lot of these is probably very difficult to find a specific answer, but do you think the fusion had already taken place by the time you get to, let's say, Homo habilis?
yes, 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 yes.
Okay, okay.
Yeah.
Australopithecus afarensis?
So help me out here. I think they occurred after the split between human and chimpanzees, right?
Right, yeah, in the human
Yeah.
line, yeah. I think about
So
4 million years ago.
yeah, so oh yes, actually you're right. Australopithecus is three-ish, 3 to 4 million, I think. Wish Erica was here. She'd probably rattle these, numbers off the top of her head. But see, this is where I don't know. Like, I'm pretty sure the hominin it probably would be, but as I said, I think based on my knowledge, which is very limited, I'm not sure how we use chromosome fusions to date ancestry. So I'm not sure about that. So all I think we can say is it must have happened after the split between humans and
Right,
chimpanzees, which is
it
about
had to,
6
yeah,
million years ago.
it had to.
It could have happened 200 million, 200 years after, or a million. Could happen during Australopithecus. So I'm not sure when that actually occurred. So it's hard for me to kind of say, did it, when you ask the question, did it happen with Australopithecus? I don't know. I don't even think
Okay.
we
No,
know.
and those are fair answers, I appreciate it. So, basically, you have this one hominin, we'll say hominin, who
Yeah.
experiences this fusion, okay? So that one hominin, whatever it is, let's just say even an Australopithecine-like ancestor, would basically be the ancestor of all humans today. And so that because all humans today have 46 chromosomes, you don't find anyone with 48. So it's been fixed, fixation has taken place. And so this would rapidly drive a species into near extinction. And so I would argue it would be
Well,
extremely difficult for that mistake to reach
so
fixation.
I think maybe it's okay. So we know today that that it is very possible that different deers. So if I go back to my slide, this will be a good example. If I click back to my slide on deers, um, so a red deer and a fallow deer, they can mate, they can hybridize, and they can have offsprings that are fertile today. And you can see that they have different chromosomes. So I think it is not necessary. Can you see my slides? Yeah. Okay. So it's not necessary that a different chromosome means that they they can't reproduce within the population. So if let's say one male or female red deer um, had the chromosome change to get 68. Okay, and that male deer reproduce with all its other ancestors that happen to have 70, and they'll have offsprings as per normal. Some of those offsprings may end up, with 68 chromosomes. Some of the other offsprings may end up with 70, and for whatever reason, as time kind of goes along, all of that particular deer ends up with 68, and then we start a speciation event that we kind of call the red deer. But it looks as if that whether you change a chromosome from 70 to 68, it doesn't really impact the reproductive rate. So you're suggesting that it must impact the reproductive rate, and that's why you're saying it's a near extinction event. What I'm trying to challenge you, Donny, is I'm not sure if that actually happens, because we can
Well,
see that they both can, so they can both have offsprings today. Those
right.
offsprings are healthy and they can reproduce. Like, why wouldn't that be the case in the past?
But so if we were to agree that these various deer species, I put a full screen just so I can see them. So
Yeah,
let's just
yeah,
say
of
we
course.
were to agree that these various deer species are related and go back to a common ancestor. Okay. So my position
Yep,
would say they go back to a common archetype.
yep,
Well,
yep.
4,500 years ago. Okay. So according to their chromosome numbers,
Hmm.
there's a variation in the numbers of chromosomes. So it's not like there was one fusion and then it all got fixed.
Hmm.
But with humans, every single human is 46. So now let's assume humans are related to chimpanzees, bonobos, gorillas, orangutans. Okay, but they have 48. So in the human lineage, at least, was that fusion not fixed in every single person? Because every single person on the planet has
Yep,
46, right?
yep,
No one
yep.
with 48.
So if we assume the common ancestor of human and chimpanzees had 48, and as we kind of said that at some stage after that, whether it's at the Australopithecus level or whether it's maybe at the Homo, which I can't answer that because I don't know, but then it changed. Okay, so remembering that I don't think the change or the fusion, I don't think a fusion that it results in a change of chromosome actually impacts the fertility of that species. Like I don't think that happens, right? And we can see that because right now today, different species, even the muntjacs, they can all hybridize together, and they've got such varied chromosomes. So obviously, it doesn't impact the fertility of those offsprings, right? So what I'm saying is common ancestor, 48, and after the split, it became 46 at some stage. So because it happens after the split, everyone after that split, whether it leads when it goes to Australopithecus and the hominid line, will always have 46. But the chimpanzees will always stay at 48 because that
Now,
split happened after. Does that make sense?
it does. Now, what about so this one person gets the fusion? Okay, so now they have 46. What has to happen to every other person on the globe at that time that has 48?
What do you mean? So if this guy had 46
Yeah.
and mated with, let's say, his partner that has 48, so all it is as long as in the mating process the different chromosomes can find its match. All right, so it might be one long one that they kind of stick together, and that's why the offsprings might be 46, or they might the other one might kind of break apart and connect to the ones with the two chromosomes instead of the one chromosome. That's why the offspring might end up being 48, right? So all I'm saying is, as long as those chromosomes can find each other and have to match, you're going to have fertile offspring. That kind of makes sense. Like
No, I understand that point, but you'd have one family, let's say,
yep, yep,
that has 46 chromosomes because
yep.
of the fusion.
You're
But
asking how it propagates. You're saying like
yeah,
how does it predict?
because there
Okay,
would be other people in different locations
yeah.
around the planet that would have
It's
48,
a it's a
right?
it's a good question, and so I'm gonna hazard a guess on this one, right? So this will be a pure guess. So, what I was kind of suggesting is because chromosomes don't seem to make a difference to, or at least much of a difference that we can observe. As I say, we can see muntjacs with different chromosomes reproduce, and we can see deers at least with 68 and 70 reproduce, and they look fine and healthy, right? They're not like having diseases or some sort of genetic stuff. They look all fine. Um, so therefore, um, if I were to kind of take that line of reasoning, there will be some offsprings with 48 and some with 46, right? So, and this is where my guess kind of comes in. There obviously is some sort of fitness benefit on having humans with 46 chromosome than 48. That natural selection has kind of pushed more offsprings to have 46 rather than 48. So eventually, over time, 46 became the number that overtook all that, basically replaced all the 48. So that's how
Right.
I understand. Now, do I know what that fitness thing is? No, I don't. I maybe there's a study on this one that kind of showed it, but it would seem logical that if there was some benefit to having lower chromosome over having a high chromosome, at least in the human side, that natural selection would basically kind of favor that 46. Does that make sense? That's how I would explain it. But like I said, pure guess, just based on my limited knowledge.
So
Yeah,
so basically with natural selection helping to
mm,
propagate
yeah.
the 46 those with 46 because there's some kind of benefit you're saying. The ones with 48 over time would basically have to be weeded out. They'd have to
Oh,
die off.
that,
Those
yeah.
lineages?
Well, since we don't have any 48 today, and we know that there was a split, and the split didn't happen in a population, it would have occurred in one that happened to then reproduce, and some of their offsprings would still have kept that. And I would assume some of them wouldn't. There must have been a genetic or a natural or an evolutionist reason why 46 was favored as opposed to 48.
So let's assume now we've assumed for many of these points that evolution's true. Let's assume that special creation's true. Let's assume there was a flood bottleneck 4,500 years ago.
Yep. Yep.
Would there not be a more parsimonious explanation in the creation model? Because the flood has what? 8 people, right? Noah, his wife, their 3 kids, Shem, Ham, and Japheth, and their 3 wives.
Mhm.
Okay, so you have a human population that's small. They're not spread out yet. They don't spread out for another roughly 3 to 500 years, let's say. And so they're all situated in one part of the planet, one geographical location. So if there was a fusion, wouldn't it be easier to explain how that fusion could have been propagated into all people in the creation model rather than the evolution
It
model? What
it
are your thoughts?
it may be, but you're still gonna have the same issue around why 46 was being selected. So if you're suggesting that Adam had 48, and because of something that happened, maybe it was a strong environment, maybe it was just so catastrophic that it damaged damaged the genome of Noah's sons. Okay, because I think Noah would be he didn't have any kids afterwards, right? So he'd be 48, I imagine. So his sons would probably be 48 as well, I imagine, because they were living during the period of the flood. So something that was catastrophic that happened during the flood caused some sort of mutational damage or change, I guess, to the son, so that when they had more children, some of them would have had 46. Okay, and I'm guessing not all of them, because some of them would have 46, so some of them would have been 48. So the question then will still, I mean, it's a smaller sample, so the difficulty would be obviously less, but the same issue kind of still occurs. So what would have made, if we go with that creation model, Noah's sons' kids would have 46, and some of them would probably have 48. What would have selected the 46 over the 48, so that as time goes on, it's only the 46 that's left, right? So I think the issue still remains, whether you look at it from a creationist side or whether you look at it from an evolutionist side. Though I do grant that with the creationist side, it's a smaller problem because you don't have, I don't know how many people, millions of people on the planet. You've got what, 8 and however many kids, or the kids, however many the kids were. Because I would suggest that if you're saying it's a Noah's flood that caused the event, it wasn't Noah or his kids; it's their kids that would have the 46.
Okay, but consider this, Ian. What if the fusion happened prior to the flood? Since, for sake of argument, we're assuming the flood happened for now, we're assuming there was a flood bottleneck. What if the fusion occurred prior to the flood bottleneck, prior to the flood, and it had not spread to fixation though in the pre-flood world? Okay, so only certain subgroups of the pre-flood population would have had this unilateral mistake, others didn't. But one group or family that did have it was Noah's family. So you have a near extinction event where now you're left with 8 people. Everybody else dies off during the flood. Who goes through this bottleneck? Noah and his family. So
Yeah.
now it's fixed in the post-flood population. What are your thoughts on that if we were to consider that, Ian?
Yeah, okay. So let me just put my head in the thinking space of a creationist. Look, if I was a creationist, that could make sense. A couple of things kind of springs to mind. So what you're saying is it is the actual, because it's the chromosome 2 fusion happened in Noah's lineage, and Noah's family was basically the only ones, I suppose, that survived the flood. That could be a rationale as to how
Yeah.
that kind of propagates support. And look,
Because now it's
I think
basically fixed,
yeah,
right? The whole
ma-
family
ma-
has it.
makes sense. Makes sense. So two questions. One, I guess we'll have to explain on a creationist view how it actually went from 48 to 46 prior to the flood, and potentially that could be, I guess, no, Adam's sin, I guess, because I think that's one of the things that you guys kind of say, genetic entropy, the fall of man, Adam's sin, and then things obviously would have gone from a perfect genome to something that's not as perfect. So I guess that could be an explanation, but you need to have some sort of explanation as to why Noah's family had it.
Yeah,
Okay.
I would
Um,
argue, and I understand you reject largely
no, no,
genetic entropy, but
yeah, but But that's how you would
I
explain it, is what I'm
right,
saying.
yeah, because I would say fusions are outside the norm,
Hmm.
and therefore fusions would be manifestation of genetic degeneration. So sometime after the flood, because of mutation accumulation,
Hmm.
because of genetic degeneration and mistakes, a fusion could have taken place because we do see fusions.
Yep.
For example, in nature, we do see fusions. You've provided examples on the screen. Okay, we see it in equids, we see it in various deer species. So we know fusions happen. Okay, so it's not outside the realm of reality. But the question I would have is, did a fusion happen in the human lineage? Is the human chromosome 2 the result of a fusion? And what I would do to answer that question is compare the purported fusion site. Tell me if this makes sense, Ian. Speak to this. I would like to determine whether or not there was a fusion in the
Hmm.
human lineage. I would love to compare the fusion site to the fusion sites in, let's say, these deer species.
Chimpanzees. Oh, I see. I see. Um, okay, so let me just put a pause on that question because I was going to say that apart from trying to explain how the fusion occurred prior to Noah's flood, and it could be very well with Noah. I'll make a comment there, and then I'll have another question. Then we'll come back to kind of what you've asked, if that's okay. So the comment is, I'm not sure if a fusion of a chromosome is actually what I would term genetic entropy, because that would suggest that someone with a fused chromosome is possibly worse off genetically compared to someone who's not. But when you look at the deer species, the red deer is like, red and the fallow deer are probably like the two most common deers. It's not like the red deer is suffering with lower chromosomes and not propagating its gametes kind of going forward. It's obviously a very successful deer. So I'm not sure if the fact that an animal with a fused that has a fusion in its chromosome is actually worse off or has genetic entropy. So that's more of a comment. Maybe they have, but it's not something that I can observe looking at deers. And
It
I
looks
would
like
have.
people, well, I think it's fair points, good points.
Hmm.
I do see people saying, because of the way meiosis works,
Hmm.
when you have a fusion event, then the offspring will likely have decreased fertility. So reproduction still can take place, but there would be decreased fertility. So would that be a bit of
Well,
degeneration or
so
a negative
so
effect?
so I mean, it's hard for me to kind of believe that, only because we've seen our population explode. Whether you think it exploded from 4,500 years ago, you're saying that you from 8 people, or however many kids that Noah's family actually had, it exploded to the 8 billion that we had today, right? That seems to be quite a rapid growth for something that's 4,500 years old. So if you think that that is decreased fertility, then I would have to assume that if it wasn't a fusion, how much worse it would be. It just sounds like
Fair
we are
point.
not seeing. So that's that point. My question then, to extend in that, is, it's a reasonable explanation of what you're saying. It happened prior to the flood in Noah's family. So we've got to then account for the fact that Noah's sons' wives probably don't have 46. If it's Noah's lineage, then I get that that's 46. Their wife, unless you're also saying then the chances of that having two separate families having 46 would be very, very low. But likely what will
So
happen is
how would that work if Shem, Ham, and Japheth,
yeah,
they
well,
have
their
it,
wives, they have it, but their
but
wives
their
would probably,
wives don't, so they
yeah,
have kids together.
yeah. So
What
their
are the
kids
number
will.
of chromosomes the kids would have?
Well, I think it would depend. I think it would be. It depends on which where which where they will kind of fall, because it's kind of 50-50 split. So I'm assuming that
Right,
50 would
50.
have 46. And then it comes down to, okay, unless you're assuming that something happened that 100% of the kids had 46, which could be, I mean, I don't know, but that could have happened. I just don't have any data that says that that has happened, but that could happen.
At the end of the day, by the time, and you're making some good points, and I like these kinds of technical discussions where we can dig deep on the issue. And so basically, by the time people groups spread out from the Tower of Babel 300 to 500 years later, the fusion, that unilateral event, would have to become fixed, because
Mhm.
once people spread out to all parts of the globe, now
It'd be
they're going
hard.
to start accumulating their own independent
Yeah.
differences and
So
alterations
I
in
think
the genome.
something has happened. I think whether it's my explanation on the evolutionary kind of worldview, and my explanation is essentially it's it's possible that some evolutionary pressures had selected for 46 over 48. Over time, 46 became prevalent. You're suggesting something slightly different, but you're also going to have a view of where the children's probably going to have 46 and 48, and between that getting off the boat and to the Tower of Babel, that 46 must become prevalent as well. But so the mechanism of selecting 46 versus 48 is still the same in both our worldviews. It's just that you have a smaller population but a smaller time to do it, whereas I have a larger population and we can do it, for a large amount of time. So
Right, more
like I say,
time.
Chromosome Fusion 2 is, as I kind of said earlier, is neither.
Agnostic.
Yeah, it's a bit agnostic. You can kind of have two different worldviews, right? When we have conversations later on about some of the genetic side of it, I would
I guess
suggest that.
that's what brings us back to, because what you're saying is so good, and I appreciate it, is the question, is the chromosome 2 fusion argument in humans, that is,
Mhm.
the purported fusion, is this completely agnostic? And what we were talking about earlier was the lack of specific heterochromatin in human chromosomes,
Mhm.
whereas your chimpanzee and gorillas and other great apes, they have that specific
Yeah,
heterochromatin.
yeah,
And so that might help us to differentiate between the explanations, where
yeah.
it goes from agnostic to now discriminatory. Ian, what are your thoughts? I know we're kind of circling back to something we already
Yep. No,
discussed, but it's important.
that's okay. So, like, I'm not an expert in heterochromatin, which I think I've explained to you before. But from looking at what it is, and there is heterochromatin in both apes in terms of in humans and also in chimpanzees and gorillas. All right. So what you're saying is that there's actually a block of heterochromatin that we can actually see in chimpanzees and gorillas, but it's completely missing in humans. So
Right.
I mean, they all have it, but there's a specific block that's actually missing in humans. And your question is valid. Well, if it's missing, then how can there be a common ancestor? Because you would think the common ancestor would have it all. And if humans came from that common ancestor, then why doesn't it have it? So it might be kind of worth kind of explaining to the audience, layperson's view. And so please jump in, Donny, because you seem to know this more than I do. But there's a couple of functions for heterochromatin. The first one is actually it provides it makes the genome more stable. So it provides, like, it's got these blocks of repeated kind of change that kind of keeps the genome stable. So it has that kind of function. The other function, though, which is more important, is that it acts as a gene expression or gene regulation. So if
Yes.
there's something that a chimpanzee is doing, the heterochromatin will be encouraging it to kind of do it more, right? So the only thing I can think about is why it could be different, and it's a pure guess because I don't think we have any papers or studies that show this. So this is a guess from Ian, but I'm pretty sure that we don't have any papers, and it'd be like a really good field of study to kind of expand. But my hypothesis would be, as we kind of split from chimpanzees and gorillas, as human kind of splits from them, the that specific heterochromatin that's missing from humans but is in chimps and gorillas is obviously helping them express something that's helping their fitness in their environment. That's something that humans don't need. Now, I don't
Yeah.
know what that is. So that's an explanation, but it's a bit of a gap in explanation because I can't tell you what that expression is because I don't think we have done that study. But that's an explanation as to why it might be
Well,
missing.
right. It would make for a good research project for you
Yeah.
guys. And so what you said is so important, so significant, I believe,
Yeah.
because one of the several functions associated with heterochromatin
Hmm.
is its role in maintaining the structural integrity of specific chromosomal regions. So
Yep.
the stability of these chromosomes, right?
Hmm.
So if the structure and function of chimpanzee chromosomes differ in many ways, I understand they're similar in many ways, but if they differ in many ways, which we know they do, then that specific block of heterochromatin that's present in chimpanzees, but not in humans, could be functionally specific to that creature, and
Yep.
therefore lend the separate ancestry model that I argue for a very specific line of evidence. Does that make sense, Ian?
No, no, it does, and there is one bit of information as well. I'll share with you. This unfortunately doesn't really lend, doesn't sway or point strongly in what in your worldview versus my worldview, because you can argue it both in different ways. But so I had a look at this, what that actual heterochromatin was actually called that's missing, right? And I don't know what it stands for, but it's actually called STSAT. It's called the STSAT heterochromatin, which is
Subterminal satellite, I think.
satellite repeats, yes, but there's a specific one that they then have in brackets, so they have the actual code itself. So it's like GATATTTCC, that's
Right.
the name of it, right? That actually repeat is what we're talking about. You can only find that in chimpanzees, you can only find the gorillas, you can't find it in humans. So, what I'm saying is that, well, it's possibly because of the environment.
Here's a question I have. Is it
Yeah.
in bonobos?
Well, what I was going to say is it's actually not in orangutans. So
Okay.
it's actually found in chimpanzees and gorillas, but it's also not found in humans and orangutans. And I didn't find any reference to it in the paper for bonobos. So I don't know, but I assume that bonobos and chimpanzees are so similar that it probably is found in bonobos. But the
Yeah,
paper
because
doesn't.
I'm wondering, and I appreciate you bringing that up,
Yeah,
because
it
the
doesn't point
model
either
I
way.
hold
It's the same.
to in terms of separate ancestry
Hmm.
would not say that chimpanzees, bonobos, gorillas, and orangutans are all related through common ancestry. I would have multiple created kinds within your great ape family. So I
Yeah.
would have humans as their own kind,
Mhm.
chimps, forgive me for my voice, Ian, chimps and bonobos. I'd
Mhm.
have them going back to their own common ancestor archetype.
Mhm.
And I think the genetics supports that.
Well, they're definitely the closest chimpanzees and
Yeah.
bonobos.
And
Yeah.
then gorillas is their own, and then orangutans is
Yeah.
their own, the couple
And
species
and
of so
and
I just want to say this because and that's why it's curious
mhm.
to me, if bonobos have that specific heterochromatin subterminal heterochromatin, these blocks, I've got it up on screen here. Let's just double check.
Yep.
Chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal
Yep. That's it.
so ST heterochromatin. And so if bonobos have it, that makes sense if they belong to the same kind as chimpanzees.
I would suggest that they would have it just because they're so close genetically to chimpanzees.
Yeah.
I would be surprised if they had it. So like I said, the fact that it's not in
If I could interrupt you, but
orangutan,
Ian, you're saying orangutans don't have it.
they don't have it. No.
Okay. And so
But it
and that
fits
would
your
fit my
worldview.
model because
Yeah.
if I'm
But it also
arguing
fits. Yep.
yeah, because so
Sorry,
if
go
I'm
on.
arguing orangutans belong to a separate created kind,
Yep.
then your chimps, bonobos, that so basically what you're saying is this type of heterochromatin species specific, which I appreciate. So
Well, it's because it's a gene expression, it's probably to do with the environment. So I think chimpanzees and gorillas live in more lowland areas. I'm not sure about humans. I guess it should be fairly similar, but orangutans are more forest. So I don't know whether it's the environment that kind of selects it. So from my worldview, it could be the environment that selected it out. From your worldview, you could say that orangutans and humans were created separate, and chimpanzees or gorillas, if you want to put that in, were created together. So it to me, like I said, that little bit of information doesn't point exclusively to
now what
one or
if
the
I
other.
said chimpanzees and bonobos,
Mhm.
they can be traced back to their own common ancestor and
Yep.
archetype,
Yep.
but gorillas, because if I were to bring in the Y chromosome, now I don't want to turn this into an entire discussion on the Y chromosome, but you mentioned earlier that it's the suite of evidence. It's I think the word you used or we agreed upon was the network of evidence. So if we bring in a network of evidence, for example, here's a visual, okay, from a secular publication on the Y chromosomes. And
Mhm.
it's very interesting to me because the way that these are laid out is very similar to the way that I lay out. And I've got a separate slide, but for sake of time, I won't pull it up. I've got my own phylogeny on how I lay out the created kinds. And then from there, I make testable predictions and see if they fit. Here, you've got the human and the gorilla. Their Y chromosome, you can see here, is more similar. Actually, here's the heterochromatin. This is interesting. So I want to do this for you, Ian, so you can see better. So if you look to right here, so heterochromatin, a densely packed piece of DNA. And so this is present in abundance in the human Y. You can see that. But in the chimpanzee, basically absent as far as I can see. Here in the gorilla, and then your orangutans. So this would fit nicely with a created kind model that says humans are their own created kind. Chimpanzees and bonobos go back to their own kind. Gorillas, they're unique. They're independent in many ways. And then your orangutans. So that seems to fit with the Y chromosome data, but also circling back the heterochromatin. Do you find that interesting, Ian?
Yes, yes, I do, but I think I might be coming to a. So I'm not an expert in the Y chromosome, but if you put that slide back up again,
Oh, sure.
from just glancing at it, I actually think they're all different. So I think you're saying the chimpanzees and the bonobos are similar because maybe it's similar in size from what I can see. Because when I look at the banding, it's pretty much completely different. And I'm not sure what the banding on the colors mean, but I assume it's like chunk of DNA that maybe is similar across the chromosomes. I don't know. Like why is it gray and dark gray and black and
I
white?
think chimpanzee and bonobo Y chromosomes are about 65%. There's a new paper, Erica and I debated it for a couple hours. There's a new paper that came out, if I remember correctly, May
But why would you say they're
2024.
the same? Like, so you're saying that the common ancestry, you got the two there between chimpanzees and bonobo. Just looking at this graph, is it because they're both similar in size? Is that why you kind of say using the Y chromosome that they should be put together? I'm just curious as to
No, it's a really good question. It's a really good question. So I could pull it up, but
because
in,
orangutans not
in
too dissimilar. If I look at the orangutans, they're
yeah, if you
not
look
too
at the two orangutans, they're quite close.
No, but orangutan
Chimpanzee,
is bonobo. The
yep.
gap, if you're saying the difference is size, then I would think bonobo Y in terms of size. I mean, I might be wrong, so correct me. But if you were comparing chimpanzees and saying that's the same as bonobo because it's a size, because I can't see what else it could be given that the banding to me is very different. But if you were to say it's a size, then I would say bonobo Y chromosome size seems much closer to orangutan Y chromosome size than it is to the chimpanzee Y chromosome size. So I'm just curious to how you came to the conclusion that it's the chimpanzees and the bonobo was created. That was a created kind. Does that make sense?
It
Like
does
I can
make
see
sense.
humans is very different because it's like twice the size of chimpanzee. So I can see why you would conclude that that might be created differently. I'm just curious as to why you would say chimpanzees and bonobo, but not bonobo and orangutans. Given that if it's the size and not the banding, then it looks like it's more similar. And I'm also assuming this is the scale. It might not even be the scale. I don't know.
No, it's a good question. So your chimpanzee and bonobo differences are largely due to gene loss. Okay. So there has been some gene loss, which is readily accounted for. But when you bring in the human Y chromosome, you're not only looking at gene loss, but you're also looking at the addition of genes or genetic content. One type of genetic sequence or material in the Y chromosome is a palindrome, and that is largely missing in I've got the section of it from the paper. And I think it's the chimpanzee lineage. So basically, bonobos and chimpanzees, I believe that we can account for the differences from an arc ancestor, let's say 4,500 years ago, because we're just accounting for gene loss. Once you bring in the human Y, it's gene loss and gene addition, but a lot of gene loss. So human and chimpanzee Ys are only about 27% the same when you consider overall architecture, gene content, size differences. chimpanzee and bonobo wise, they're about 65% the same, but the greater portion of those differences is in the world of gene loss. So I don't know.
So, so
Does
I,
that help
I,
you?
I it does, and you're reinforcing also what you said before, which is why you think humans are separately created versus chimpanzees and bonobos. I get that, but I was asking about bonobos and orangutans. Like, what would be like?
Okay.
What just looking at that, whether you say orangutans is a gene, what did you call it? Like gain. It looks like it wouldn't be that much of a gene gain, only because it doesn't look like, if this is the scale, that much different compared to bonobo and chimpanzees. It looks like the gap between bonobo and orangutan is either the same or less than the gap between bonobo and chimpanzees. Like, couldn't you also argue the same thing? But why did you choose chimpanzees and bonobo and not bonobo and orangutans? Because that's almost the same size.
Well, if I'm understanding your question correctly, chimpanzees and bonobos, we would both agree go back to a common ancestor. But once you bring in orangutans, then the common ancestor for chimpanzees and bonobos and orangutans goes back. You can see
Yep.
here,
Yep.
Borneans,
Hmm.
Sumatran, however you
Yep.
say that, goes back. Let me see, my mouse is freezing. Goes back to 13 million years, right?
Yep.
So gorillas are closer, according to the
Yep.
evolutionary model,
Yep.
but the gorilla Y is more similar to the human Y than
Hmm.
either is to the chimpanzee and bonobo Y, right?
Yep.
So right there, I would argue is an inconsistency in the hierarchy. So therefore, we would have to rule out the orangutans as being part of the same created kind as chimpanzees and bonobos, right?
Yeah. So help me with your understanding of this. Like I said, I'm not very familiar with Y chromosome, so I can maybe try and do a bit of study before we have a conversation next on this one.
Yes.
But as my understanding is, the way we kind of track back and use genetics to confirm nested hierarchies was we don't really look at Y chromosome, but my understanding of Y chromosome is that it's not a good way to look at it because it's subject to a lot of change. Like, I think people even are arguing whether the human Y chromosome will eventually just evolve itself out because it's like changing so rapidly that it might actually
Mutation
just
rate is
disappear.
high.
So given the fact it changes rapidly, maybe it's not the best indicator. As I understood it, it was looking at pretty much the entire genome. So it's comparing things like, and I know we'll discuss this in the next podcast, but it's looking at shared ERVs. It's looking at shared line elements. It's looking at shared ALU elements, and the percentage of shared is how we kind of draw the where it kind of sits in the phylogeny tree, and we make an assumption
Are,
around
are you
molecular.
saying that you would look at very specific genetic markers that would make sense in
Well,
light of
even if you look at the entire ALU elements that sit in humans, compare that with the entire ALU elements that sit in chimpanzees. I think from what I could see, we know that 95% of humans' ALU elements are shared with chimpanzees, and something like 91% shared with gorilla, which suggests, if you're accepting of the common ancestry model, that we diverge from gorillas much earlier than we diverge from chimpanzees, only because of the shared ALU. And it's not just ALU, it's line elements, it's sign elements, it's a ERV. So that's I think how we kind of
okay, so
build up.
basically you would look to so if you look to a genetic unit
Yep,
like an ERV or an ALU sequence,
yep,
then you have humans closer to chimpanzees
yep.
when we compare the
So
entirety of those DNA elements
yeah,
than we do with
even more
humans
than
and
that,
a gorilla. But then if we
yes,
look to the Y chromosome, okay, in terms of that DNA, that's a uniparentally inherited DNA compartment.
mhm.
Then we have humans and gorillas close. So to me, it seems like, and feel free to challenge this assertion, it seems to me that we're dealing with cherry picking. Oh, well, we like the ERVs and the ALU sequences because it fits the expected hierarchy, but we don't like the Y chromosome because it doesn't fit the expected hierarchy.
So the question would be, when you have a bunch of data that seems to fit. So even forgetting just ERVs, and even if you look at the protein coding sequences, and I know Dr. Jeffrey Tompkins will have a different take on this, but if you just look at the protein coding sequences, you can compare the similarities between human protein coding sequences and chimpanzees, which is much closer than it is between human and gorillas and so forth. If you look at pseudogenes as well, and you look at and track back whatever pseudogenes that we have, whether it's the one that gives us that vitamin C deficiency or whatever, you can track that through lineage. So it's entire human genome comparison. We can pick out an element of ERVs and compare that, and it just so happens to match if we picked up the ALU segments elements or the line or the pseudogenes or even the protein coding genes, they all will match. So then we have another data point, which is Y chromosome, that doesn't seem to match. So the question would be, what is the explanation as to, instead of like throwing, like taking the Y chromosome and throwing the rest away, because it's not just it's entire genome sequence. So it's actually a proper comparison of the entire creature's genome that seems to match and fit a nested
Yeah,
hierarchy. But you've got
and,
the
and
Y
and that's a
chromosome. So
fair.
the question is why? And I think the explanation is that Y chromosome changes so rapidly that it's probably not the best way to compare. So as I understand it, that's the explanation that I've
Okay,
heard.
yeah, and that's a fair point. So I've written some stuff on what you're saying here, and I don't want us. I do want to bring us back
Yep.
around to
Let's
where
save
we
that
were
for
on the.
podcast two.
Yeah, yeah, exactly. Let's say that on podcast too. But I would like to say in terms of the non-coding regions versus coding.
Hmm.
So on the screen, I've written a bit on this.
Hmm.
That so your long non-coding RNAs.
Hmm.
Actually, if you look to the previous point, protein coding genes are the building blocks.
Hmm.
Okay. And so those protein coding genes, I would
Hmm.
expect to be more similar between creatures because they're your basic building blocks for these creatures. But then your non coding sections of the genome, they're more organism specific. And so those genes provide the metadata for the building blocks that are the protein coding genes to function. And yet that's what we find is once you bring in your non-protein coding genes, Ian, then we find less similarity. But the
Yep.
protein coding genes are, and I agree with you, and I think that's a fantastic point, they are more similar
So
between creatures
uh,
rather than being taxonomically or more species specific.
yep. And if I can make two observations on that, the first one being, yes, you're right. If you're just looking at protein coding genes, a human and a chimpanzee would obviously need many of the same kind of tools for survival. All right. So you'd
Right.
expect that to be the same. But as I understand it, it's not just that the genes are the same, it's the location within the chromosomes that are very similar. Right. So given 6 million years of evolution, there are slight changes, but where it kind of sits on the chromosomes are very similar. In fact, it's as I think Eric has done the work, and I know Dr. Jeffrey Tompkins disagrees, but I also know that Dr. Robert Carter has also replicated Eric's work,
Yes.
and like so,
And Dr.
so
Carter's doing some good work on it too.
yeah, so it's not exact, but the protein coding area is so similar. I think the number is 98 to 99% similar, right? So there's a little bit that's different. Now, when you bring in entire genome comparison, and there are different lengths and all that kind of stuff, but when you account for that, that number drops to something like 95 and 96%. So yes, you're right, Donny. There are differences, but it's still extremely similar. There is a 5 to 6% dissimilarity between humans and chimpanzees when it's looking at entire genome comparison. But if you look at protein one, it's 96, 98 to 99. So
Now, let
you're
me
right,
say
there is
this.
differences,
No,
but
and
it's
that's a good point.
it's not huge.
So when it comes to these similar sequences,
Yep.
even between humans and chimpanzees, let's say, it's very interesting to note that when we look at gene expression and gene regulation, right? So gene regulation basically controls the timing, the location, and the amount in which these genes are expressed, right? That's why you hear down regulation, up regulation. So it is a very important mechanism. And you'll find in those similar sequences, Ian, differences in how they're regulated and also how they're expressed. So even in the similar sequences, there are still differences, which I find to be fascinating. And that
Yep.
would reflect separate ancestry expectations. Does that,
Or
Ian, does that make
or
sense?
yes, it does. Or that the genes were in the common ancestor was to do one thing, but because of the environment, it needed to adapt. So it won't be doing exact same thing as what it
Right.
needed a chimpanzee to do. So like I said, you could argue both ways. It just
Yeah.
depends, just all the data. So I take your point. It's not 100% exact, but when you do the entire genome comparison, it's 96%. That's
Are
pretty
you
high.
basically saying, and just for the audience, are you basically saying, so let's assume now evolution's true. Okay, so if we were to go back to the common ancestor between humans and chimps,
Mhm.
those similar sequences, they'd be identical also in gene regulation, gene expression. But since the split, there's been some alterations
Yeah,
because those two lineages are independently changing.
and that's why it's not a human and chimp is not 100%. That's
Right.
why if you look at protein coding genes, it's 98 to 99. If you look at entire genome comparison, and they do, I don't know exactly the mathematics that sit behind it, and Erica kind of showed it, but she accounts for the different sizes, etc. as well. But it's not 100%. It dropped that 99% drops down to something like 96%. So there is and we're talking about 3 billion base pairs here. So it's not like one or two genes. So it's still hundreds of thousands of genes that are different. So it's still quite a big amount, even though it's 96%. But the similarity is very high. So that accounts for, I think, the sizes of the genome, the different repeats and deletions and all that kind of stuff. There's a methodology that geneticists use to account for that. That is used in the program, I think, that Erica kind of put together, and that's the one that Dr. Robert kind of also replicated, and kind of said, well, he doesn't think Dr. Jeffrey Tompkins did it correctly, and he, I think, said Erica did it correctly, and he came up with the same number as Erica, the 96%.
Ian, let me show you this quickly. I do appreciate those points. And then we should let the audience know. Okay. So we are going to be doing an ongoing series. Consider it a podcast, a discussion podcast. Me as a creationist, Ian Chen as an evolutionist, and we are going to dig deep. So this is an ongoing podcast where we are going to really dig deep on these topics. We're going to keep them digestible about 2 hours. Okay. So we're going to start wrapping up this one, but then we're going to continue and we're going to discuss all topics related to ancestry. So I just wanted to let people know that, because I do know people enjoy these conversations where, rather than formal debate style, we actually get the opportunity to examine these papers, to discuss the technical aspects of the creation evolution controversy. And I think this is a good way for us to advance in how we, from different worldviews, can understand these issues. So this
Mhm.
is from the 2024 paper, just to kind of land the plane on the Y chromosome topic, and then get back to the chromosome fusion
Mhm.
issue. So if you pull up the visual here, the complete sequence and comparative analysis of ape sex chromosomes, if you compare here, let's say you take, okay, so here's your human Y.
Mhm.
Okay. So you take your human and you can compare it to each of your great apes. Remember, your model would suggest that humans are human great apes. Your other great apes are non-human great apes. So
Mhm.
humans to human, 100%. Of course, that's what
Mhm.
we'd expect. That's how we know it's working.
Mhm.
Human to chimpanzee, it's only 26.2%.
Mhm.
Now, earlier you're asking about the orangutan and the bonobo. So take the Borean, I think it is, Borean orangutan. So take
Mhm.
this here,
Boy, yeah.
compare it to the same 100%, and then take it here, compare it to bonobo. See, it's only 28.6%. So at least according to my model, yeah, that fits created kind expectations. But if we compare the other orangutan, so orangutan to this specific species, 100%, and then compare it to the other one, 67.4%. Okay. But what's interesting, because there are regions that don't align, right? So if you actually compare the borean,
Why are they different? Sorry, can I ask a question? So
yeah, it's a
if
good
you
question.
look at if you look at bonobos, like just the very left
First,
of the table, If you look at bonobos and chimpanzees, it says 59.6%. But then you skip over to chimpanzee, so the next one, and you go chimpanzee, but no, why is it 65.3 there? Shouldn't it be exactly the same?
Right. It's a great question. I'm 95% certain that's because the Y chromosomes don't all align. So if I'm going to take the bonobo and compare it to the chimpanzee, because with this paper they have a really sophisticated way now of comparing the Y chromosomes where they have very small amounts of just meaningless data. They've got a really good sequencing. So because they don't align, I think is what it is. So then if I were to just take the chimpanzee and compare it to the bonobo, then there's going to be differences.
So
But
you're saying,
we could
yeah,
yeah,
it still
go
doesn't
ahead.
quite make sense because if it doesn't align, there's obviously some sort of algorithm that says we will drop out the sections that don't align, right? So whether you're comparing bonobo with chimpanzees, that algorithm that says, well, we'll drop out these unaligned sections, there's still going to be the same sections that are unaligned whether you compare chimpanzees with
Mhm.
bonobo, vice versa. Like I just don't understand
But I
why. I
think it's
mean, they're
because I think
obviously
it's the reference. So like whatever your bonobo references that they have in the database and then the chimpanzee reference, Y chromosome in the database, it's going to vary depending on which one you're mapping onto
so
that
so the
reference genome.
bonobo
I could
bonobo
be
that
wrong. It's probably something rather than making an error on this, we could probably just look it up after.
yeah,
I
okay,
think it
let's
has
do that.
something to do with
Um,
that. The
can I make
reference.
an observation here? So
Yeah.
let's look horizontal. So let's just pick one. Let's go. And it's I use horizontal because it's easy for me to compare X and Y by doing horizontal. So if you go down to the third row and you've got human as
Yeah.
that one. So I'll go horizontal because then you can see the X and the Y, and I'll explain why why I'm doing it this way. But you can see the X and Y much easier by doing it horizontal, right? So yes, okay. So X chromosome, that seems to be in line with what's we think
Yeah, they're all
genetically.
very uniform, the X
Yeah.
chromosome.
Well, I mean, human compared to chimpanzees is much higher than human compares to bonobo, and but also much higher than gorilla, which kind of supported what we kind of thought, which was the split between chimpanzees and gorillas. Gorillas much earlier, so that kind of makes sense, right?
Mhm.
Now, obviously, when you go to the Y side of it, the numbers are much lower. Okay, so that's a problem. Okay, like why is it much lower? So the question, but when I look at these, like even though it's much lower the differences still are in line with the X. So when I compare human with chimpanzees, and it's 45.1%, which is really low. Like you think, wow, it's more than half difference, but it's still higher than humans with gorilla at 23.2%. And the bonobo is closer to the chimpanzee, so that kind of makes sense because we
Right.
say that so the
And we would
relative.
both agree that they're related, bonobo, chimpanzee.
Yes, yes, but what I'm
Yeah.
saying is that whilst so it's to your point where you kind of say, why is it so different? Like, why is the
Right.
Y chromosome so different? My explanation is, well, I've heard it's because the Y chromosome mutates very fast. So
Yeah.
whilst it mutates very fast, it's still mutating fast, but relatively the same hierarchy is what the X is saying. So when you just look at the Y, okay, it's mutated so fast that it's only 45.1% comparable between human and chimpanzees, but that's still higher than gorilla, which is kind of in line with what the X is saying. And bonobos are higher than gorillas and but lower than chimpanzees, which is actually still what the X is saying as well. And the numbers are bigger, but the relative differences are in line with what we're seeing with the X. Does that kind of make
But if
sense?
you were to go, yeah, it does make sense. But if you were to go with, let's go with the smaller number. So human to chimpanzee,
Hmm.
26.2%.
Hmm.
That right there, as we were discussing earlier, those are some massive differences because it's not only gene loss, it's gene loss in the sense that we have entire pieces of DNA that has to be lost in the human lineage, like that specific type of heterochromatin, the subterminal block of heterochromatin, and gene addition. But if you look at the bonobo and chimpanzee Y chromosome, so here's chimpanzee up to bonobo 65.3%. It's still close enough, I believe, where we can both agree in our models that they go back to a common ancestor, because largely what we're dealing with here is gene loss. And if the mutation rate is as fast as we understand, it's about 2 to 3 per generation in humans. And I'm assuming it's just as fast if not faster, in your great apes, then those differences can be accounted for, especially if you have massive deletions, right? Of just a whole block of the Y chromosome, then it can be accounted for. But again, once you bring in the chimpanzee, human, and bonobo, now we have more than just gene loss. So that's where I think the problem is for the proponents of common descent versus the separate ancestry advocates.
Yeah,
Does that make sense?
yeah, it does. I just it probably doesn't help that I don't understand the difference between the columns and the rows. But even looking at the columns, which is where you've gone to, human and chimpanzees
I'll
seem
say
to.
this in the commentary papers, the
Yeah,
authors are going with the smaller numbers. So if you read the
yeah,
commentary papers, those they're saying surprisingly humans and chimpanzees are only 26.2%,
yeah,
or gorillas and humans are 23 points. So they're going with the so what we're looking at here is a range, but
yeah,
what I see in the commentaries or the feedback articles, layperson
yeah,
articles, they're going with the smaller numbers. So that
yeah.
seems to be the more
And
important one.
I guess what I'm saying is, let's just go with a smaller number, but don't the numbers itself, like, isn't when you do, let's say, human and chimpanzees at 26.2% using the vertical smaller number, and then you
Mhm.
go gorilla and humans at 23.2%. Well, okay, those numbers are much smaller than what we're seeing in the X chromosome, but the fact that the gorilla and human is lower than chimpanzees and human is exactly what we see in the X chromosome. So it's not so much the fact that it's smaller, it's that the up and down is actually in line with what we expect on the X chromosome. So then the question is, why is it smaller? And that could very well be not positive, but could very well be because the Y chromosome is known to be rapidly mutating compared to X chromosome.
What I like to say, Ian, is, and I'm sure you've heard me say it before, it's the differences that make all the difference. So when it
Mm,
comes to creation versus evolution or universal versus separate ancestry, we can both largely account for the similar sequences. We can account largely, both models, for homologous patterns in morphology, anatomy, and genetics. But it is the differences that I argue the separate ancestry model can account for. So the common design proponents, we can account for differences and similarities. You guys can account for differences to an extent. But once we get to these massive differences, what I typically see is untested hypotheses. And hypotheses are good, but without the testing, then isn't it at this point just a story? Or how do you view that? Like, do you find those
Well,
explanations about faster mutation rates
so
and
well, I'm not so I'm limited in my knowledge, so I'm not sure whether we have tested the fact or understood why it's smaller. So I know that paper came out and it was surprising. I get that. How long ago was that paper? Was it like 10 years
I do
ago?
want to represent it accurately, and I recommend people check out my recent debate with impromptu debate with Erica for a couple hours on it. They tested it in the sense that they looked at sperm competition in chimpanzees, mating habits,
But
polygamous
so but that,
relationships.
that's the kind of so we make a hypothesis as to why it's lower, and it's because we think the Y chromosome mutates faster, and then when we test it, it's because of sperm competition. So I get that, right. So maybe that's the reason why. So that I mean the question then is, do you not think that's valid? That sounds like if it's been tested, and I haven't read the paper, so maybe it hasn't. Maybe there are some flaws in the testing or whatever, but it sounds like a reasonable hypothesis. Um, that it's the Y chromosome that mutates faster, and the reason why it's faster is because of sperm competition or whatnot. Is that has that
So he,
does that not make sense? Or
well, it's a proposed solution, but where I think the issue arises, and right here, so for example, I put a slide here and I wrote down one of the major issues that I think exists here is the differences between chimpanzee and bonobo Y chromosomes reflect diversity and structural rearrangements, not gene content. When you bring in the humans, okay, let's say we were to go with sperm competition. But the thing is, we're not just looking at large scale deletions, gene loss, but we're looking at the addition of various forms of genetic content, right? And so I think that's where the challenge is for the evolutionary community at this point, is to explain, okay, bonobos and chimps, we can both agree go back to a common ancestor because we're largely looking at gene loss. Bring in humans, you got gene loss and the addition of genes like palindromes. Very,
Are you suggesting that it's much more common to see gene deletions than it is to see gene additions?
yeah, yeah. Well, let
Because
me ask you.
I don't know that I don't know if that's true. Only because I know when, for example, when and my knowledge of this is just listening to what Erica kind of went through with chromosome fusion too, was that she was able to account for what she calls indels, right? Which is basically we know when
Insertion, deletion.
yeah, yeah. So it sounded to me like it's like a gene insertion. So the extra information that you're saying, it didn't sound like that was a very rare occurrence, and it actually happens. Now, it might happen a little bit slower than what a deletion would occur, but it didn't sound like it was a rare occurrence. So
But then it gets into an even bigger debate, which I'd love to discuss in
Mhm.
our next segment on this, is what is the I'll put your slide up for now as we start to wind things down. There's two very different explanations for the origin of DNA differences and genetic diversity, right? Ian, between our models. You would argue or assume that the vast majority of DNA differences are the result of mutations over time, where I, as a creationist, would argue that the vast majority of autosomal nuclear DNA differences are the result of created heterozygosity or design diversity. So, where you would say indel insertion deletion or gene duplication, you're looking at a section of the human Y chromosome or the human chromosome, whatever chromosome it is, and the chimpanzee chromosome, and you see a piece of DNA in the human chromosome or genome that doesn't exist in the chimpanzee genome.
Yeah.
Let's take the Y chromosome, that specific location is missing. And so you would assume that that's because of a deletion. But maybe it was always that way. Maybe that DNA difference is
Yeah.
not the result of a mutation, right? A couple
So as
assumptions
as
there.
as I understand it, it's not just saying here's the sizes and one is less, so it's a deletion. It's we're looking at the elements within it. So let's say it's AA, and I know it's CATG, but let's say CCAATTGG, and then in the chimpanzees we see CCAATGG. So there's a T that's missing there. Then we look at the similarity of that gene and we kind of go, well, everything's exactly the same except that in the chimpanzees it's missing a T. So
Right.
ergo, that T is a deletion because everything else around it's the same. Now then when we look at an insertion, we kind of see CCAATTGG, and then we see the same in chimpanzees, but then in the humans we see another CCAATTGG, which is exactly the same between the two. We go, well, that must be an insertion because you've copied a gene stretch that is exactly the same as one that we saw previously. We don't see in the chimpanzees, so we can safely assume that that's an insertion. So I think a duplication, I should say. So it isn't just saying the different sizes, and we just say, well, it's less, it must be deletion. There actually is a bit of working to kind of see, is it really a deletion or is it really an
Right. There's a
insertion?
methodology.
Yep,
Right.
yep,
I
yep,
understand there's a methodology involved, but it
yep.
still would ultimately, and I guess that's where the debate would go next,
Mhm,
is that methodology does that methodology work, or are we still assuming, okay, humans and chimpanzees go back to a common ancestor, and so all these, even if it's a single letter difference,
mhm,
then the evolutionists would argue that single letter difference, correct me if I'm wrong, is due to a substitution. But as a creationist, we would say no, it's not necessarily a substitution. That could be a created difference.
mhm.
So the evolutionists assume it's a mutation. We would assume it's a created difference,
And I
depending.
think, yeah, and you're right, right? I mean, so when it's a single letter difference, potentially it's a little bit harder to tell. When it's an actual specific gene where it's multiple letters, it could be 100 KB of gene data that we saw removed. That's harder to explain as a substitution because you're substituting a big chunk, right? So I think you're right if you're only just talking about one. then it's much harder to figure out whether it's a substitution or it's a deletion or
And
whatever.
then that's where we get, it's a fair point you make, because that's where we get into this whole debate. And again, correct me if I'm wrong, when it comes to measuring or figuring out the overall differences between human and chimpanzee genomes, if you have a deletion of, I think you said, let's say 100 letters, a block of DNA that doesn't exist in the chimpanzee genome of 100, are we going to count that as 100 differences
Mhm,
or one? Because if we
mhm.
count it as one gene deletion, right, then it's only going to be one. But if we count it as 100, well, there's 100 differences. So that
Yep.
would inflate
And
the
I
amount of.
know Eric has kind of gone through it because it was one of the questions around, how do you measure the differences? Like I think the example she uses, and she tried to relate this back to what a creationist would understand. Let's say you had the same page of the Bible and the same page of the Bible and the creationist Bible, one atheist don't have a Bible, but evolutionist Bible, the same page, exactly the same as the creationist page, but there's one word that's different between the two pages. Do you take that one word as being the difference, or do you take that page, the entire page, as being the difference? Right? There apparently is some sort of algorithm that's agreed, geneticist has agreed as to how you would treat that. I can't. I'll try to refresh myself prior to this before we have the next conversation. But there was a way that geneticists actually account for those differences. Like there's a way that's standardized methodology that they use. Um, and I think that's the one of the main criticisms of um, Dr. Jeffrey Tompkins is that he used the erroneous way of calculating it.
Let's park that topic where it is. We're definitely going to come back to that and
And
start
this
to
is going
wind
to
down.
that that's going to be, sorry if I can just jump in. That will definitely be an interesting conversation
Oh, it
because
will,
if you
100%.
if you start going onto what is an insertion and what's a deletion, the logical, and I'll just say this to you because this might be something that you might want to keep in the back of your head because we'll probably discuss it later, is okay, assuming it's a insertion or a deletion, you're saying it was created that way. All right. So if it's an insertion, we must explore what does that insertion do? Like, is there
Right.
something
Function.
functional?
It'd come
Not
down to
yes, correct.
function.
Yes, and then if it's a deletion, it's that well, is that because the fall of man and everything's getting worse, and that's why it's deleted, or is it created, deleted that way? In which case, why? So we're going to get to the next level of discussion. Is to okay, let's agree that there was an insertion, deletion, whatever it looks like. We can agree whether it's one gene or multiple genes or whatever. The question then the creationist will have to explain is why. Is it like was it created that way? Then for what purposes? Or did it degrade? And in which case, was it created degraded, or why did it? You get what I mean? And so we're going to have those interesting discussions. So I guess to the audience,
Right.
stay
It's
tuned
going to
for part
bring
two.
us, it,
Yep.
it's going to bring us to what I like to call the discriminatory evidence. And so and we talked about this at the beginning of our program here. Are these nested hierarchical patterns that we see in the biological world, are they due to design or descent? And
Perfect.
so what we're dealing with in our discussion a lot of the times
Mhm.
is or are agnostic lines of evidence, like the
Yep.
chromosome fusion issue. But eventually in our podcast series here, Ian, we're going to really dig deep on the discriminatory evidence. And I think that's going to come down to DNA function
Mhm.
or non-function in the evolutionary model. I know Dr. Dan Stern Cardinal has done a lot of work on that issue. And so his videos will be great prep for you.
Mhm.
And I think we'll have a really good discussion on that
Yeah,
issue.
definitely, definitely, and perfect because that was actually the fourth point that I brought up with Kent, which is really around the DNA evidence for nested hierarchy. So, and it pointed to whether it's function or not. So, when we explore the second podcast, we're basically be exploring the next bit of evidence that I presented to Kent, which look, I'm sorry, he didn't answer. He was, as I kind of said, he's an athlete. He was running away as fast as he could. So I know you won't be doing that. So it'll be an interesting discussion with you, Donny. But can I just say, with this whole wrapping it up with this, what I was kind of trying to present in the debate with Ken was what I just
Actually, Ian, if I could,
yep,
because and I want you to get to that. I want to put this full screen so the audience can
yep,
see it better.
yep,
So this brings us back, and I think this is going to be a great place to start wrapping things up. And I want to get what you're going to say there, because we've discussed a lot about fusions in the animal kingdom, the potential existence of a fusion in the human genome. But I think we've both agreed and correct me if I'm wrong, if I misunderstood, we've agreed that fusions in general, and if you want to assume that there's a fusion in the human lineage, it's largely agnostic to the overall debate. The evolutionary
Yeah,
model,
yes,
they have their
I
explanations. We
I
could
think
have our explanations.
I think by itself, you can probably explain it in your way, and I can explain my way. I think obviously that my explanation is stronger, but I couldn't just say using fusion it would cut out exactly what it would basically discredit creation. Like as I said from the beginning, I don't see why creationists would want to die on the hill with this one because you can explain it you cannot deny the science and still explain it in a creationist viewpoint. But people like Kent, and that was my point of this. Of putting this up was Kent. Why do you deny? Why do you accept chromosome fusion? Which then he said he didn't. So I think Kent has this view that chromosome fusion does not occur, whether it's humans and apes or whether it's other animals. I think from what I understood, he doesn't believe it actually happens at all. And there's so much evidence and papers that show that it actually happens, right? So my question was, why would a creationist accept fusion occurring, and then still call these animals the one kind, but yet the same methodology of saying these fusion occurring, putting it on the ape side, then saying that apes and humans created. So that was basically my question. Like, why do you use different standards?
So how I
This,
would answer that, Ian, briefly
mhm,
is, and I definitely want to kickstart our next discussion on this
mhm,
with at least 30 minutes
mm,
where we examine, and maybe this will give you the opportunity to go look at some of the DNA data on these chromosome fusions, because, and I'm just going to read your question out. I'm going to answer it how I would. Okay. Why would, let's say, Donny
yeah,
deny
why
a
would
chromosome fusion occurred between chimpanzees slash gorillas and humans when he accepts it occurred in multiple species within the deer kind? Okay. So I do accept that chromosome fusions have happened in nature. Okay. I did a roughly 2 to 3 hour response video to one of Gutsy Gibbons' response videos to me. So just response after response after response on this issue where she brought up through Ruhif, if I remember correctly, a potential chromosome in a pig. Okay. Now I examined the pig fusion site and compared it to the purported fusion site in humans, and they look nothing alike. They were very different. The purported fusion site in the pig genome was massive, and I got the data. We can get into that in our next podcast.
are
But
you
there's a
comparing sorry, the pig fusion site with a human fusion site? Is that
yeah,
what you're?
as in like I'm looking at the signature. How many telomeric repeats are there? How large is the?
Isn't it erroneous to compare what happened in the fusion for the pig versus what happened in fusion for humans? Like they're
Well,
not.
I guess what I'm saying, with the fusions that have occurred in nature in
Mhm.
animals other than humans, what I've seen are massive signatures where there's no debate. There can be no debate made. The amount of telomeric repeats, TTA, GGG, CCC, TA, the block of DNA, the signature is so massive that it can't be denied that it's a fusion. But then when you look at the purported site in the human chromosome 2, the signature is very small. It's only about 800.
So you're referring to something like, for example, in a deer, because I'll shoot you through a paper because I've you asked for it earlier in this conversation about showing you the deer fusion, and I've got a couple papers where they actually do a comparison of, I think it's chromosome 23 and 4 on a red deer and what's chromosome 1 in a sika deer, and I don't have the sika deer here as well, but um, and they match pretty much the two chromosomes in terms of the difference of what it actually does. The telomeres kind of match, and the centromeres for the red deer is actually quite large. So I think what you're saying is, why is the centromere, as a comparison, the centromeres is really large on the deer chromosome, which obviously looks like it's two telomeres kind of joined together. However, when you look at the equivalent in the human chromosome 2, the centromere is much more reduced, so it doesn't immediately jump out like it's a two telomeres kind of added together because you would think that that
Right.
competition.
It doesn't look
Yeah,
like,
yeah,
it doesn't really look like two
yeah,
chromosomes slammed together, because I would
but
argue the signature should
yeah,
be a lot larger. That we should
we
find
we we,
more
de-
of
we
these
definitely
instances.
yeah we definitely should talk about it, but because this, and I'm trying to remember what Eric kind of talked about, but there are and you probably know it. It's the what is it? It's one of those DDX gene family. that is apparently only
Yeah, the
found.
DDX11L2
Yes, it's only found next to a telomere, but in chromosome fusion 2, it's found
in the
next to the
middle,
centromere. Yeah.
right?
So, and I think one of the things that I've heard creationists say is that there's a functional gene across the centromere, and
Well,
I'm
remember
pretty
they've named that gene though DDX11L2
yeah,
because
yeah.
they do believe that that gene is associated with what? A
Well, I,
type of
I
fusion event, right? So
yes,
it didn't
no,
name itself the DDX11L2, so one
yeah,
could say that this belongs to that family, but it begs the question, is this
yeah.
area the
So I
result of
think
a
from what
fusion?
Eric
But
was saying is that the spanning of the centromeres is not 100%. Like I think it's like 50.
telomeres to be specific, right? Like
Yeah,
your
but
centromeres would be the
yeah,
center of your chromosomes, but basically these telomeres,
yeah,
which are your end caps of your chromosome. So yeah, in the center,
yeah.
we should find a lot of these sequences, and we're going to get into that because
Yeah,
we're probably going to
let's
discuss
get
that for an hour.
let's get into it. Obviously the next podcast, but yeah. So I think I'll rewatch some of Erica's stuff, but she had some explanations as to, when you're talking about looking at the structures that kind of show the fusion. It's not just looking at the size of the centromere, but it's also looking at what's around there and in all other genes in that
Right.
particular XL. So you'll have to ask the question, why is it different in chromosome fusion two then? So yeah, if it's not a fusion event, why would that
Yeah,
chromosome
I would
look
say
like
this before we get into too much detail on it, which we will, because I had a research project, brief research project with Chris Roop, where
Mhm.
we searched the genome. We made a prediction.
Yeah.
If the so-called fusion site's not a fusion site, we should find other instances in the genome and other chromosomes
Mhm.
where we find these repeats,
Mhm.
TTA, GGG, CCC, TAA, these telomeric repeats in forward and reverse in
Yeah.
mirror form, back to back. So we went in, we didn't know, but we found them in multiple locations and we found them in a fairly large signature in
Mhm.
chromosome 9. So I wrote an article on it. I have it all
Yeah. I
laid
think
out visually. We'll go over that.
yeah, and
And
I think
nobody would say, as far as I know, that the chromosome 9 is the result of a fusion. So we do see that signature. Now, I will say that the signature in the chromosome 2 is more dense. Okay.
yeah.
The size is about 800 base pairs. But then when you compare that to a fusion in the wild in an animal species, it's very small because you can find, I think in the pig, it was like 40,000 sequences. But in the
Yeah.
human chromosome 2, it's, 798. And I understand these are
Yeah.
points we're going to go back and forth on.
Yeah, no, we'll definitely will. But and I feel like I really hope that we just can continue because I know it's late night for you and stuff, but it's a bit, and then we shouldn't keep you up, and your wife will probably hate me if that's the case. But
She
it
could
it
never.
all she'll never hate me because I'm just a teddy bear. Um, so look, it comes down to like how dense it is, right? So I mean, if you're finding CATTAC, like and that's the only thing that you find, and that's the only thing you search for, then okay. If you're finding like many, like what point do you think that those repeats are enough? So we'll get into it as well, but it depends on what you're searching for because if you have 3 billion base pairs in a genome, you're bound to find that sequence at least once, because it's just so many options. But will you be able to find it 100 times repeating? So that becomes like the signature. Like at what point is the repeating gonna mean that it's actually a centromere or telomere? Because if you find it once out of 3 billion base pairs possibility, then you'd assume with the basics of probability that you're gonna find that signature at least once somewhere. Does that kind of make sense? But you're not gonna find it repeating 100 times. Do you get what I'm trying to say? So I think yeah, it just
Well,
comes down to that.
I would agree with you that we should find. So maybe I will just to satisfy our first discussion here. Let me screen share real quick.
I go to bed unsatisfied. Never let Donny go to bed unsatisfied.
Well, you bring up some fair points. So I would agree
Yeah.
that it should be of no surprise to anybody to find these internal telomeric repeats, TTA, GGG, CCC, TAA, throughout the genome by chance in isolation. Okay. But
Mhm.
that's not what we found. We looked for them in both forwards and reverse in mirror form where they're back to back, right? This telomeric like hexamer sequence. Okay. And so we went in and here's the chromosome 9. So this is what we discovered. So here's the whole sequence, the surrounding area as well. And what you find is, okay, TAGGG, TTAGGG, TTAGGG. So back to back. So many repeats. Okay. Tandem repeats is what those would be called. And then the reverse complement. CCC TAA, CCC TAA, CCC TAA. So they're touching in the head-to-head fashion.
So
That
you
we wouldn't
found,
expect to find.
but you found this once, or how many times did you find it?
It's a good question. So we found them in. So here's the chromosomes we found them in. 1, 2, 6, of course, 2,
Yeah,
6, 9, 10, 15, so on and so forth. But the largest signature, other than the chromosome 2 spot, was this one. So this is the one I felt was worthwhile to
yeah. And
publish
chromosome two,
visually.
how big was the chromosome two one? Was it like
Great
massive?
question. So here's the chromosome 9, right?
Yeah.
Well, what's funny is the chromosome 9 is actually more pristine than the chromosome 2, but
Mhm.
it's still dense, but it's less. So it's more pristine, but less dense. The chromosome 2, which is right here, is more dense but less pristine. It's more degenerate. So there's interesting characteristics about each. Okay. So here's the chromosome 2, 798 base pairs, but you'll notice that it's a lot more degenerate, right?
Mhm.
TTAGGG, but you don't get another tandem repeat until over here.
Mhm.
But if you actually go to the chromosome 9, it's just back to back to back to back to back, which is very, very interesting.
Yeah.
It's a curious
And
case
but
there.
that's the only sequence that you've so look, look, the fact that you found, even though, so we know that telomeres degrade. I mean, I think that's the point of what it does. It's supposed to degrade to kind of protect the chromosomes, right? As it kind of replicate and stuff. That's as I understand, that's what it does. So it's designed to kind of degrade to protect the chromosome. So it's no surprise that if it becomes a centromere, that it would actually have a lot of the degradation happen over time. So that's one point. The second point, though, like keep in mind, it's 3 billion base pairs, so 6 billion of these letters, and you've shown maybe 20. Like, what's the chances of 20 occurring just by chance? So if you had shown me 2,000, that would be different. But you've shown
Well,
me,
then I could
and
say the same thing about the chromosome 2, though,
yeah,
because we find a
but
very
we
dense
expect it.
region.
But we expect it to be there, right? So and we expect it to be more degraded. So that's what you're showing me. You're showing me what is expected, right?
But
No? Oh,
no. Well, with the chromosome 2 area,
mhm,
your signatures that you should find,
mhm,
right? What we do find doesn't seem to be the expected signature because in a prominent paper associated with the chromosome 2, the authors said, why is the area so degenerate? So even they recognize that this area, if it's a fusion, is a
mhm.
lot smaller than they expected. Because again, when we look at fusions in the species that you point to, which I agree are the result of fusions, they're undeniable. They're huge signatures. I mean, you've got thousands of these tandem repeats, it's just undeniable. But here you've really only got what? You could count these up. There's really not that many. In
Yep.
terms of
It's
TTAGGG, you've
almost
got one that maybe
it's
10 to
almost
15.
it's almost like I'm hearing, like I'm not hearing it from you today, but I've heard creationists tell me the opposite. So I've heard, so what I, from my understanding, these are telomeres. They're designed to degrade. The fact it degrades actually protects the chromosome. So when it actually fuses and becomes a
Oh, I get
centromere,
what you're
it
saying,
does
yeah.
it does nothing. So you would expect it to degrade. So that would kind of make sense. But I've also heard creationists or other creationists, I think maybe even Nephi said this to me at one point, where he said, no, no, no. The centromeres have an actual functioning gene that spans the centromere. In which case, you would think if that's a functioning gene, it would be fixed. That you would not expect that level of degradation. So on one hand, you kind of say, why is it degrading? And then I hear other creations say, well, there's a functioning gene there, so it shouldn't be degrading. Like I don't know which one is true because to me they seem to be contradictory. Like they're two contradictory explanation. So it's either it does nothing and it degrades, or there's a functioning gene and it shouldn't be degrading.
Well,
So it
so
actually should be
yeah, it's great
there.
questions, fair points that you bring up. So I did check the database. You can do it yourself. You can be an evolutionist, creationist. You can just go do it yourself. And so I looked up DDX11L2. That's the
Mhm.
gene that spans the fusion site. This is when I looked it up, retrieved it, and I wanted to see what the functional associations are with it. So
Yep.
it is active, okay, and it has co-expression. DDX11L2 has close to 2,000 functional associations.
Hmm.
But remember, it's a non-coding RNA gene as well.
Yep.
So
Yep.
it's not equivalent to the type of activity that you would find in a protein coding gene, right? There's more activity there.
So it's not important. Is that why it
No,
degrades
no,
very fast? Like,
no. Well, it looks here, at least according to the database, that the DDX11L2 gene that spans the so-called fusion site,
when you say
it is
spans the fusion
functional.
site, are
It
you
has
saying
activity.
it crosses across those centromeres? Like, what do you mean by span the fusion site?
It so
Or
y-
it is,
let's so, so you've got the fusion site, right? That's
yep.
798 base pairs.
Yep.
The gene actually spans it. It goes from one end to the other. So it's overlapping the fusion site.
The entire fusion site, you're
Yeah,
saying
like
is
like like- yeah, like the fusion site is overlapped by this gene basically.
so that's a
So
big gene. That's a 768 kilobit. Um, that's a big
and
gene. That,
what's interesting is the DDX11L2 gene produces these long non-coding RNAs of various types. It's alternatively spliced. It has dual promoters. And what's interesting is the second promoter or
Mhm.
genetic switch, that's what a promoter is, of that gene is the fusion site itself.
you're,
So some have
you're
argued
saying
that
that's not a centromere that we're seeing in chromosome fusion 2. That's actually the DDX11L2 gene. If it spans whatever the letters it makes up, those letters basically define that gene. That's not a centromere. It's not the graded centromere. That's wrong. It's actually this gene. You get what
well,
I'm
the
saying? You're saying
centromere
it
would
spans,
be found in a different location. The fusion site itself is just the telomeric repeats that we're looking at.
so it doesn't
Your
span the fusion
cryptic
site. Then
centromere would be
it's
elsewhere in the chromosome. Because remember,
so
your centromeres are the centers of a chromosome, help with
yeah,
stability of the chromosome,
is
is my
which is
understanding.
only found in chromosome fusion 2 because it is the fusion of two telomeres, which we call a centromere. Because it's not found anywhere else. It's only found in the chromosome fusion 2. So I'm just curious when you say this particular gene spans the fusion site.
Yeah, well, it spans the 798 base pair region. It spans those
So,
those
so that
interstitial telomeric repeats, from my understanding.
yeah, so that repeat, that code that makes it a repeat, CC whatever, that's actually the description of the DDX11L2 gene or whatever. It's not actually centromere. If you're saying it spans the
Well,
centromere, it's
it
actually not
could be a second promoter. That region of sequences could be a second promoter or genetic switch of that gene, the DDX11L2 gene. But I'm not necessarily concerned with that, because the more interesting point to me is the fact that we found a very similar signature, a more pristine signature in the
yeah.
chromosome. Well, I should ask you, do you believe the chromosome 9 is the result of an ancient fusion? I'd imagine not, right?
No, but my question is simply, if there's 3 billion base pairs of numbers, what's the chances of this sequence, which is not a big sequence, appearing randomly?
I would say it's a very low chance if there's a higher chance if so. For example, there's been papers that you can look at in the secular community, in the conventional literature, where these internal telomeric repeats, the TTA, GGG, the CCC, TAA, are actually not necessarily associated with telomeres. They're functional DNA units that help in gene expression. So if what
So,
we're looking at in the chromosome 2 area and the chromosome 9 are simply functional DNA units that are necessary for the health of that gene or chromosome, then that makes sense. I think that's why we shouldn't be as surprised that we find so many of these throughout
so the only
the
thing I'd say
genome.
to that is, okay, so you're assuming this red and this blue is some sort of functioning gene sequence, but I don't think you can find that in the centromere. I think from what you showed before was that it was broken in some way, right? So if you go to that chromosome fusion 2, I don't think that stretch you can find exactly. If you're saying that stretch is functional, let's say that's true. Where can you
Well,
find that stretch
are,
here?
are are you asking, do we find this exact 798 base pair
No,
sequence anywhere?
I'm saying when you scroll up to chromosome fusion 9,
Yeah.
yeah, this one, and you're saying that that is actually a functional gene or a functional sequence of some sort, and let's say that's true. Can you find this exact? And then you say, well, therefore it's functional in chromosome fusion 2. From what I can see, I can't see this exact stretch in basically here what you're showing me here. I can't. So
Well,
if that sequence is functional, great. Let's just say that's functional, but I can't find that stretch in chromosome fusion 2 from what you're showing me. So ergo, I can't see why you would say chromosome fusion 2 has that functional gene when I can't actually see that gene stretch in what you've just shown me for chromosome fusion 2. I've seen broken parts of it. That's what you're showing me, but I can't see this exact one which you're saying is functional in there.
it's a fair point, but the portion of the chromosome that you want to focus on, the important portion, is the signature, the TTA, GGG, the CCTAA, your telomeric repeats in forward and reverse, the reverse being the reverse complement,
Yep.
in a head-to-head arrangement,
Yep.
because that is supposed to be indicative of a fusion. And that's
Yep.
what we find in the chromosome 2 are these telomeric repeats in a head-to-head manner. That should only exist in the area that is the result of a fusion, but we find it the same signature elsewhere in chromosomes that aren't the result of a
Then it
fusion.
goes back to obviously in chromosome fusion 2, it's like it's a massive amount. Like it's so much bigger, right? So that would make sense.
But
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 then
but well, I want to
also,
say this, Ian, but that's relative because it's a massive amount compared to other similar regions with these head-to-head telomeric repeats in the human genome. But if you compare it to a fusion outside of the human genome in, let's say, a
yep,
deer or an equid species or a pig,
yep,
then
yep,
now no, it's very small. So
yep.
it's relative. It depends what you're assuming it's large compared to,
It's
right?
I know we're kind of going way over time here, but so then when I asked, well, how is it not just chance that that appeared? I think you said it's because it's probably functional. In which case, I said, well, if it's functional, why can't I see that? That description of what is functional. Why can I not see that in the centromere? So
Uh,
ergo, maybe it's functional, but it's not a functional
What do you
gene.
mean by centromere, though? Your centromere is not in
It's
here.
isn't that the fusion of two telomeres? Isn't that what that
Yeah,
is?
yeah. Well, so it'd be your telomeric repeats, but
Yeah.
your centromere would be found elsewhere in, like, I've got some slides. So the cryptic
Okay.
centromere would be because two active centromeres would be detrimental. So if there's a
Mhm.
fusion, one of the centromeres would have to be deactivated. So they've looked for that in chromosome 2, but it's outside of that region that we're looking at.
Okay, okay, let's park it because I'm going to have to read up on terminology. But I thought a centromere was when two telomeres join together, and it was only found in chromosome fusion 2.
What you find in what's commonly called the fusion site, those are your telomeric repeats. But if you look for the alleged cryptic centromere, it's a totally different sequence. So maybe I'm not explaining my, so it's a different, it's what's called an
I'll
alphoid
yeah.
repeat DNA sequence.
Okay, let me look up terminology. Like I said, I had thought based on my simplistic understanding, you've got the ends of chromosomes are telomeres,
Right.
and those telomeres fuse together. Okay, and when they fuse together, you still have those two telomeres joined together, but happens to be in the center of this new chromosome, and it's called a centromere. Now, the repeats are the sequences that make up the telomeres. So when they actually join together, those repeats will be repeated across those centromeres. So that's what I thought they were almost the same thing as a result. You're saying they're not. That obviously means I don't understand this. So give me some time to understand this for our next conversation. But that's what I thought a centromere was. And the repeats were just how you described the telomere. Does that make sense?
Yeah. Well, a centromere is basically the region of a chromosome. Its function is basically in cell division, from my understanding, helps the cell divide during that process. But where you would find those alphoid repeats is different than where you'd find your telomeric because your telomeres are your end caps of chromosomes, but your centromere would be located differently. So I'm not sure if that makes sense, but basically I'll put it this way. Ian, there's two different signatures we'd be looking at. So there's your telomeric repeats, but then there's also your centromere, and that's made up of what's called alphoid repeats, I think it's called.
Yeah.
So every chromosome has a centromere, but in a chromosome that's the result of a fusion, there should be a second one. But if it's the result of a fusion, then that second one should basically be deactivated, because two active centromeres would be detrimental to that.
Okay, that makes sense. That makes sense.
Yeah.
Shouldn't it be three then? If every chromosome has a centromere, and the one that's been created is basically the two telomeres joined together, you still have the centromeres from each of those two chromosomes together. Does that make sense? So there should be
Yeah.
three.
Well, you'd have two chimp-like chromosomes basically come together. They'd fuse together, and so you'd have an area with a high amount of telomeric repeats.
Mhm.
But you'd have two chromosomes coming together, so one centromere per chromosome, and therefore you'd
Okay,
have
okay,
one
yeah, it's
active
okay,
one
yeah.
and then one
So
deactive.
yes, so I had the wrong terminology, so that makes sense. So let me just familiarize myself with the terminology. We can have a discussion in the next one. Definitely
Yeah,
will be interesting.
do that, and hopefully we can do it sooner than later, because
Yep.
I do have a whole section on the cryptic centromere and where they
Yep.
think it is. And so if you read up on it or maybe watch some videos, then we can our second part, we can start touching on the
Yeah.
centromere issue.
Sounds good. Sounds perfect. So yeah, so if I can summarize your position is basically you do accept this fusion that occurred, but you believe that the fusion that happened between chimpanzees, gorillas, and humans, at least from my worldview, is actually different enough that it's actually not the same fusion that we see happen in other animals. That's basically your position.
It's a great question that you have. Yeah, yeah. I see that your screen shared right now. That's important that we're here.
Yeah.
So basically, I have no problem with a fusion in the human lineage.
Hmm.
Okay, because I do agree fusions happen, and fusions happen in species that belong to the same created kind, like in equids and deers. But I want to see the evidence in the human chromosome too, that is as strong as what we find in these animal species. Does that make
Hmm.
sense? So I'm okay with there being a fusion. I'm just not
Yeah,
currently
I
completely sold
I'll,
on it
I'll
being a fusion.
I'll have to familiarize myself with Chromosome Fusion 2 for humans, but I don't see why the methodology on how we know it's a fusion between different animals and humans are going to be much different. Like I would have thought they would have compared the same thing, which is essentially, telomeres, those repeat sites, and all that kind of stuff. I thought like, well, I don't see why the methodology would be different. So I'm pretty sure if we went through in detail the chromosome fusion two, and we compared to how we did it with deers, with canines or whatever, I don't think you're going to see that's going to be much different. Does
Maybe
that make
what
sense?
you could do, Ian, is, and I can do it with you, we can help each other, maybe chat about it privately. Let's pull up these papers. I do have one on a pig
Hmm.
species where there's apparently a fusion there. And let's line up the fusion sites. I'll say the purported fusion site in the human chromosome 2, and then the fusion
Hmm.
sites in at least one of these species, and let's compare the number of repeats, the tandem repeats. Let's compare, let's see the size of this, because again, a prominent paper on the human chromosome 2, the authors asked, why is the area so degenerate? So they recognize that it's a pretty small signature,
Hmm.
and I'm suspecting that the signatures in these creatures, Ian, are very large, like probably thousands of sequences. That's my, I'm going to predict. Maybe I'm wrong. This will be good to look up, but I suspect that the signatures here, why it's so undeniable that these are fusions, is because the signatures are massive.
Hmm.
And maybe that's
Okay.
something we can both look into for our
Yep.
next discussion. What do you think?
Well, depends. Do you want the next discussion to be an extension of chromosome fusion two, or and the third
Yeah. Why
one
don't we
be?
kick off the next one
Yeah.
on this issue, so we leave no stone unturned, and then from there we can dive into your next slide and topic,
Yeah, which is more ERVs, ALUs, and all that kind of stuff. Yeah, no
and then
problems. We
do
can do
this till the end of the world, because I have fun, and I enjoy talking to you. You're a good guy, and I appreciate your
that. Does Kent
willingness
think I'm a
to
good guy?
study these issues.
Does
What's
Kent
that?
think I'm a good guy? Does Kent think I'm a good guy?
He does. Yeah, he invited you to Dinosaur Adventureland. He wants to have an
Yeah,
in-person debate. So,
that could be just to make sure I
you
never.
guys are going to be best buds in no time.
I might not be able to come back from that though.
That's
Who
true. You
knows?
could be banned, but at least we'll get that fourth debate. Well,
Yes.
Ian, let me you and I will continue talking backstage, but in terms of this recording and this discussion, we are
Mhm.
wrapping it up. And I hope you had as much as fun as I did, you and the audience. And okay, God bless all.
Thanks, everyone.